Good morning, guys. Please let me know in the comments if you are here for the day. Okay, you should have noticed at this point, they're going to tell you, that I have posted the marks. So you should be able to check your mark at any point. I would look over it and just make sure I haven't made any errors. So if there is something on there where you're like, oh no, I actually got this on this assignment or whatever, just drop me an email. It doesn't happen very often, but I do occasionally make mistakes, and I don't want to shortchange anybody. If I accidentally put it in a number that's too high, I probably wouldn't say anything about it, but it's up to you. It would be very honest of you to mention that. That happens too sometimes. So, but anyway, I wouldn't fault you. I am sending out reports today at some point. They're fairly general. Because they include data from 2 units, as you know. They are basically an overview of, are you completing the homework, which I have from your homework portfolio? What is your current mark in the course? And do you owe anything in terms of assignments? And that's pretty much it. It's not a super in-depth report. So anyway, you'll be getting a copy of it as well. I send it to you and your parents. You can expect that it's exactly what you think it's going to be based on what you have just seen in your mark report. So I don't think anybody's going to be surprised at what's in there. But generally things are going well for this class, I should say. My other class is having a little bit of more trouble than you guys are. I'm not sure exactly what's going on over there, but they're having some trouble completing stuff. I mean, the course is basically exactly the same, so I don't think it's the course that's different, but for the most part, you guys are doing a pretty good job getting stuff in on time. There are a few students that are struggling a little bit with timelines. Again, if you're letting me know that you're going to be handing stuff in a couple days late, a day late, a couple days late, that's fine. That's fine. I don't even mark that stuff late as long as we are agreeing on a reasonable extension. It's students that aren't honoring the extensions that we're discussing, or are ignoring them, or not even discussing extensions and then handing things in quite late. That's when we get into an area where I don't believe that you're managing the course appropriately, and I'm really concerned with you getting the credit at the end. Because keep in mind, when we end, there isn't like several days where you can just catch up at the end. Pretty much when the course ends, I mean, we do have those support days, instructional support days. I think there's 2 at the end of the squad. I have to double check the schedule. But I'm writing my report cards during those days, guys. Like those are emergency days. Those are days that I can support you if you owe me a thing. So what's happened in the past, I'm not trying to freak anybody out or anything, but in the past what tends to happen is that students that are handing things in a week late or 2 weeks late, we get to that point in the course and they owe me 3 or 4 things, and there's just no chance. You're just not going to get it done, or you're going to complete it and throw it in, and it's going to be terrible. It does not demonstrate a clear understanding of the stuff that you did this quad, because you're like a month behind in real time. It may only be like 4 or 5 days behind in the course, but that's a month of normal course time, and you just can't do that. It's too big of a burden for most students to make up at the very last second, and so then those people don't end up getting the credit. Like the reason that I'm pushing people to communicate with me and make reasonable extensions for themselves and meet those deadlines that they're making is that I want to keep you on track and I want you to be successful. Okay, I'm not doing it to be like, oh, I'm not trying to set up artificial barriers here for you where it's just like, well, this is the due date and this is just, there's no reason for it. There's a reason for them. They're there to help keep you on track. That's the purpose of them. I mean, they're also in order for me to give you some kind of timely feedback on the stuff that you're submitting. But beyond that, if you're not adhering to them, you're basically removing your chance of being successful in the course, which I don't want. I want everybody to be successful. So like the deadlines are not there for just like to create this artificial difficulty for you to get things in on time. They're just to help keep you on track. So just try and keep that in mind. I'm not. They're there for a reason. And if you're going to be proposing an alternate deadline, you need to meet your alternate deadline that you are making with me. So make a reasonable one. So for the vocabulary lists, maybe where do you find your marks? Look at they're posted on the front page of the course. So let me show you. Yes, cat. Oh, I forgot to feed the cats. Hold on. If I don't feed them, they're gonna cry the entire time we're on here. One sec. Sorry about that. To answer your question, Kim, yes, that's exactly what I mean. Like, what is the importance of that term within the context of that unit? Why is it important for us to learn about phenotypes? Why is it important for us to learn about the concept of non-disjunction? So I'll give you an example. Okay, I'll write out a complete definition. Try and give you a helpful one. I think I provided an example in the actual assignment itself. There's an example listed that includes an importance. But let's say that we're talking about the concept of non-disjunction. Okay, I'm gonna type out one right here. And this is not a dictionary definition. Okay, this is me giving my understanding of what non-disjunction means. I mean, and I guess it's a little bit different than you giving your understanding because I'm teaching this course, but bear with me here. The screen is black. Oh, yes. Like the backdrop? Yeah, I'm not typing this on my iPad. I'm typing it in the chat. I'm assuming you don't mean the entire screen. The entire screen's black? I don't think it is. Okay, so that would be my definition for non-disjunction. Okay? Non-disjunction is an error that can occur during meiosis during anaphase 1 and anaphase 2. In a non-disjunction error, all the chromosome material in a tetrad in anaphase 1 or in a pair of sisters in anaphase 2 is pulled in a single direction and not separated properly. This leads to gametes being formed with too much or too little genetic information, otherwise known as aneuploidy. Non-disjunction is important to study in genetics because it's one of the primary causes of genetic illnesses that stem from the meiotic process. It can profoundly affect how genetic information is passed on to offspring. And I spelled 2 wrong there. Sorry. So I'm providing context for why non-disjunction is important as a thing. Why did we even talk about it? Why did we learn about the idea of non-disjunction? Because it has such a profound effect on how genetic material can be passed on to your offspring. That's why it's important within the context of the unit. Okay? So like not every term is going to have this like profound meaning, but you can't use as your meaning in your vocabulary list. Like it's important because it helps us understand genetics. Like that, a lot of people did stuff like that, and that's too vague. So you need to give me a little bit more. Why is it important to understand the meaning of this term and to use this term? What knowledge does it give within the context of whatever unit? Okay, and so for that unit, it was within genetics. For this unit, it's going to be within the context of understanding animal physiology, so structure and function. So why is it important to understand that term in terms of understanding the structure and function of animals. Sure, something kind of along those lines. Why, it's not necessarily why is it important within the animal, but why is it important that we're learning about it? Why are we incorporating that vocabulary as part of our understanding of physiology, which is structure and function within organisms? And specifically it's animals. This is animals unit. So why are we learning about this particular structure and function? What does it add to our understanding of physiology? I don't know why they don't just call this unit physiology. Animal structure and function is such a weird... that doesn't make sense to me to why not just call it physiology? That's what it is. The structures and the functions of those structures within animal systems. Like there's a word for that already. I don't know. Sorry, hopefully I answered your question, Kim. Yes, it's the air server background. That is correct. It's because I was typing it in the chat. I get that, Serena. I'm typing it in the chat. Okay, I'm hoping I think I answered all the questions here. If not, don't be afraid to flag me down. I kind of answered it. Uh-oh. You're explaining the reason we're learning about the term. For example, like you chose the term. Basically, you said this is an important term in terms of understanding it within the context of this unit. Like you chose it, right? You guys are picking the terms that you want to use in your list. They're intended to be terms that are core terminology, important terminology for the unit. So all you're saying is just why. Why are you saying that this is a core piece of terminology? What now that you understand what the word means, or you're clear on its meaning, why is that helpful for you to know that? Sure. I could run through any one of these notes. Let's pick the circulatory system note that we just did. I mean, I don't know what terms are new to you, but pulmonary, for example. Okay, pulmonary means of the lung, or you could talk about pulmonary circuit as a full term. The pulmonary circuit is the circuit of blood flow that goes from the heart to the lungs and back to the heart again. The pulmonary circuit is important within the context of physiology because most animals have a circuit that is only responsible for the oxygenation of blood and for dropping off carbon dioxide. Or I guess you could say it's a common feature for most animal circulatory systems. That's because oxygenating blood and removing carbon dioxide is one of the main functions of the circulatory system. It also is a direct connection between the circulatory system and the respiratory system. It's where the two systems interface. Is that satisfactory, Cam? Yeah, I mean, that's both, right? That's explaining why animals have that system and also why we're learning about it. It's both of those things. Because you're right, that is the why the animal has the system. But we're discussing it because that system is a key system in order for animals to survive. They require a pulmonary circuit that oxygenates blood and removes carbon dioxide. And it's such a key component of the circulatory system that there's an entire half of the heart devoted just to pumping blood through that system. It's a key system. Okay, sorry. Okay, hopefully I've made that clear. Okay, I'm going to stop talking now. We'll keep going. All right, so yesterday we were primarily talking about the circulatory system, and I believe we're on day 14 now. So what we're going to cover today is we're going to talk a little bit about control of the heart. So how do you make the heart go? Why does it keep going? And we're going to talk a little bit about the electrical activity in the heart, how you measure that electrical activity, what it means, how you increase your heart rate, how you decrease your heart rate, and then we're going to spend a little bit of time talking about some illnesses of the circulatory system. Okay, so that's what we're going to do this morning. I have also posted a fetal pig dissection. Now you are supposed to be able to see this part, so my apologies. Let me throw this up quick. So, yep, we're going to talk about this stuff. For the illnesses component, there is a slideshow again that you can access yourself. I've posted a link to it right there. Like I said, we're going to talk about the electrical activity of the heart and reading and understanding the electrical activity. That's called an ECG or an EKG, depending on which country you're in. In Canada, we usually call them ECGs, electrocardiogram, spelled with a C. In the US, they use EKG, where cardio is spelled with a K. Now, I think the reasoning for that is that the EKG, the cardiogram, was a German invention, and cardio is spelled with a K in German. And so I think that it's just they've just kept that spelling of electrocardiogram. I'm not really sure why. But anyway, in Canada, it's usually called an ECG, but in the US, they call it an EKG. But it's the same thing. So you don't need to watch this video. This is an enrichment video, but it talks a little bit about more detail about reading an ECG. We're going to talk about it in very general terms, like what the bloops and bleeps are on the EKG. But this goes into quite a bit more detail in terms of reading an EKG. If you're interested in that, I'll leave that totally up to you. I think it's fascinating, but certainly you may not think it's that interesting. So then you can skip it if you're not interested. I also normally at this time in the course would do a fetal pig dissection with you. So I would get some people. Well, normally you would get your own fetal pig, you in a group of 4. And you do a dissection class. I'm really sad about this stuff, that we missed this stuff, because this stuff is like the meat and potatoes of biology. But well, anyway, we are where we are. I do one. In fact, it's my fetal pig dissection from last quad. So we did it in the hybrid classroom. I had some students come up and help me do it. So it's a recorded version from last quad. I wonder if this will play on here. Will it actually work? Is it going to play? Not sure if it's going or not. Oh, yeah. So it does work. I do some cutting. The students do some cutting. We're going to go through, and it's basically a review of all of the systems that we've just talked about. So we go and identify the structures of the respiratory system. We go through and identify the structures of the circulatory system, some of them that we can find in the fetal pig. That circulatory system's a little bit tough. We definitely do the heart. And we go through and identify the structures of the digestive system. So it's a little bit of a review. It shows what it looks like in an actual organism. It's not it's not laser clear because, well, I'm doing this on a desktop here with a webcam, so it's not perfect, but it's not bad. You can see most of the structures. So if you're interested, you can watch this as a review. I recommend it as a review before you sort of get into preparing for the quiz. But I suppose it's optional. You don't have to watch the fetal pig dissection. Normally you guys would do it, and then we actually do a formal lab with the fetal pig, where you like record like some stuff that you've learned. You do some biological drawings and stuff like that of the pig. But again, the course is different now, so I'm not going to concern myself too much with that. If you're curious as to the procedure that we're following, if you are planning on following along with it, I made a little link there to the fetal pig dissection in the textbook. I more or less follow the procedure of the fetal pig dissection in the textbook, and that's the procedure that you would follow if you were doing the fetal pig dissection. So anyway, that's available to you if you want. Normally I always do it, but I don't have access to a fetal pig right now. I'm not supposed to go to school. I emailed and asked, but they were like, oh, don't come to school. You're not really supposed to come to school. So I was like, all right. Also, my wife really doesn't want me to dissect a pig in my house. So, which I kind of understand. It doesn't really smell the best. So anyway, that's where we are. I have a video from last quad. The second half of the day, I'm going to leave up to you to work on your medical technology presentation. So that's where we are with that. Keep in mind that this is the last day. Oh, I swear I corrected that. It still says before midnight on Monday. Do I have to reload this? I swear I fixed that yesterday. Oh yeah, I did. I did fix it. I just need to reload it. Anyway, this is the last day of the unit. So tomorrow we're going to begin the plants unit. That means that all of the assignments from this unit come due tomorrow at midnight. So that would mean your medical technology presentation, your vocabulary list for this unit, and the quiz. Okay, so the quiz should be available now. I'm hoping it's available now. I can see it there, so it should mean that it's visible. And you can write it anytime between now and tomorrow at midnight. Technically, you can write it after then, but I would try and finish it by tomorrow at midnight, or it's going to stack. And again, my recommendation is that you write it during Learning Block 3 so that I'm available. You can just jump into the Google Meet and say, hey, I'm looking at this question. It doesn't make sense to me. I don't understand this. And I can just tell you. I can just clear it up with you right away. If you get outside of Learning Block 3, it gets a little bit more difficult for me to provide support. I'm not like always sitting at a computer here, obviously. So anyway, I'll leave it up to you. But that's my recommendation. There's a time limit, but it's, I think I wrote it on here, this 2 hours, but it's like a minute, two and a half minutes per question or something like that. It's quite a bit. So it's not really a rush, but you do need to be familiar with the material. So again, it's written to be open book. It's written to be still a challenge, even though you have access to your notes. So you need to get all of your notes ready, and you should review them. You should be preparing for it like you normally would prepare for a quiz or a test. And if you do that, I mean, you probably should be fine. Based on how people have done on the previous quizzes, people are in general doing very well. I would say that the averages are exactly where they are normally for unit tests. So somewhere in the neighborhood of 78 to 82. That's an average mark for a unit test that are for this course always. So the online quizzes are no different. The averages are always falling in the exact same range, and so I don't think that the quizzes are harder than a typical unit test, but I don't necessarily think that they're just a breeze either. Some of you are doing extremely well. Good. I mean, if you're doing extremely well in the unit quizzes, it means that you are prepared and that you understand the content. So good for you. If you're struggling a little bit with the unit quizzes, feel free to drop by during Learning Block 3 and we can talk about some study strategies. I'm happy to chat with you about some ways that you can better prepare for them to help get you that mark if you're trying to improve your mark. Okay, one other thing to mention about marks, just really quickly here, is that people are concerned at this point where their mark is, no, I shouldn't say people, some people are. If you are one of those people, just be aware that there is lots of opportunity for you to improve your mark still. Okay, there is 3 quizzes, including the one for today. There are 3 assignments, including the one that you're going to submit today or tomorrow. And then there are also 3 vocabulary lists, including the one that you're going to submit today or tomorrow. That's a lot. It's more than half of the content for the course. And so, could you increase your mark by 10%? Absolutely. That's certainly possible to do that if you are improving stuff as you go. And so don't panic if you're not really happy where your mark is currently at. You can absolutely improve it still. It's not like I'm giving you this mark at midterm and saying, well, this is where you are. It's not going to move from here. So, I mean, obviously that's not how it works. So anyway, some people have emailed me and asked. It's still early in the course. Let's talk about the circulatory system. I'm spending too much time going on and on. So we left off talking about the three main types of blood vessels: arteries, veins, and capillaries. Does anybody have any questions from yesterday's homework, by the way? The stuff from the textbook? I think most of it was straightforward stuff. Some people asked some questions about the partial pressure stuff from the morning, and those were good questions. Those questions are not the most straightforward. I'm going to keep going, and then I will circle back if anyone types anything. I realize that there's a big delay, so I'm happy to go back if anything pops up in the chat. But just if you do have questions, I'm happy to go back and talk about them. I want to spend a minute right now talking about how you actually send a signal through the body. And we talked about the idea of hormones. So one way you can send a signal through the body is you can have a cell. The idea of partial pressure, yeah, the concept of partial pressure will be on the test. I'm not going to ask you to calculate a partial pressure or anything like that. That's for chemistry. But the idea of like what happens to the rate of oxygen transport into the blood as the pressure decreases in the air, the partial pressure of oxygen. The idea, the concept of that it would decrease in how fast you can perfuse oxygen into your blood as the pressure goes down. That concept of that is something that you should know for the test. Yeah, I'm not going to ask you to do any of the math. That's for chemistry. Hopefully that answers your question. If you want to send a signal in the body, you can have a cell over here that releases a chemical, whatever. A chemical is just a bunch of shapes, basically, a protein. And then that would get taken up into the blood, and it travels through the blood and the circulation. And then there's going to be another cell somewhere that is intended to receive that communication. And it'll have a receptor that's specifically designed to interact with that hormone. It'll bind it, and then the cell on the other end will be able to receive that signal. So that's hormonal communication between cells, or chemical communication. And there's a delay, obviously. That's not instantaneous. So this is fine for a lot of things. If you want to send a signal from one cell to many cells, this is what you want to do. If you want to send a signal for a long time, Zoe, I'm not sure. Did I answer your question? Hopefully I did. If you want to send a signal for a long time, this is what you want to do. If you don't need your signal to be instant, this is what you want to do. However, sometimes you do want your signal to be instant. Okay. For example, let's say that this cell here is a cell in your eye. Okay. So we've got some. we've got the cells at the back of your eye. Like in your retina. Okay. This is a very rough drawing here. Okay. And you see boy. So some days I got the examples ready to go, and some days I don't know what I'm drawing. Let's say you see a lion. Oh no. Oh no. Oh, God, I'm not an artist. This is supposed to be a lion. This is a very derpy lion. Oh no, I should just go out of my head. Okay, this is a lion, guys. It has a tail. You see a lion. This is the kind of signal that when you receive this, you need to react very quickly. Or, it could be also not necessarily seeing. It could be you touch something that is very hot or something like that, you're going to burn your hand. Anyway, any kind of danger signal. Okay. So the light from this is going to travel very quickly at the speed of light to your retinal cells at the back of your eye. And then they're going to send a signal that's going to go along a nerve. It's a very short nerve to your brain. And I'm going to do a absolutely terrible job drawing the brain. That's supposed to be a brain. So the signal is going to go from your optic nerve to your brain. And then your brain is going to process that signal. And then it's got to send that signal out to lots of different spots in your body. It's got to tell your leg muscles to move. It's got to tell your heart to speed up. And so that signal is going to go down through your spinal cord. Just another nerve. And then it's going to go out through your peripheral nerves from your spinal cord to your leg muscles. And it's going to send a signal out through your vagus nerve, which is going to go down to your heart muscle. It's going to tell your heart to speed up. And I'm going to draw probably the worst heart ever. Probably should use some different colors here. I'll do the the signal transduction in blue. You don't have to draw this, by the way. This is just illustrating my point here. Okay, so you got to send a signal to the brain. That signal is going to get distributed in the brain. Then the brain is going to process and send a signal down to the heart and send a signal down to your leg muscles and all this stuff, right? This has to happen instant. I mean, there's obviously a delay of milliseconds, but we're talking about an entire process from beginning to end that could be done in 100 milliseconds. Okay, a really, really fast response. It does not take long for all of this to occur. If you did this with hormones, the response that you would get here would take tens of seconds, not tenths of seconds. Okay, and so obviously you need a faster signal transduction mechanism. And you'll notice that the method for sending these signals around is nerve. Nerve is how that's to send a fast signal. Okay, so I want to really talk about in a very basic sense, and you spent a whole unit on this in grade 12, so I'm not going to go into super detail here, but I want to talk very quickly about how you send a signal along a nerve. So nerve cells have one end that receives a signal, okay, and then they have a really long tube, which can be extremely long. This could travel like the entire length of your arm or the length from your base of your brain all the way to your tailbone. These can be extremely long cells. Okay, so this would be one single cell, and then you've got another cell on the other end that is going to be the cell that communicates with neighboring nerve cells. And so it has these little structures called dendrites, and they are for communicating with the cells around it. Okay, and there's a nucleus up there. So this thing has got to be able to send a signal really fast from one end of this thing to the other. And it can do that in milliseconds timelines, really, really quick timelines. So how does it do it? We're going to zoom in on just a little section here of the nerve. And again, you don't need to draw this. I'm drawing this for illustrative purposes, but I want you to get an idea for what is actually happening and how this is associated with the concept of electricity. Because I'm sure you've heard this before. The idea that your nerves operate electrically. And the same thing is true for your heart, by the way. That's obviously why we're touching on this now, because the heart has an electrical system as well. This goes back to, like, we have had a very basic understanding of this for at least 250 years in science. This idea that there's some kind of electrical activity going on in the heart, and this used to fascinate scientists. It is one of the motivations for the writing of Mary Shelley's Frankenstein, if you've ever read Frankenstein before. The idea that you could use electricity potentially to animate the body. So anyway, this has been like a sort of a scientific fascination for a long time. But if we zoom right in on this thing, if you actually look at the dendrite, you would see that if you zoom in on that nerve cell, you would see that there are these little pumps that go across the surface of the cell. So these are going through the cell membrane, okay? And there are different concentrations of ions on either side, okay? So you may remember what an ion is from grade 10 chemistry. What's an ion? Throw it out there. So, someone take a shot at this. You do basically a whole unit on this in grade 10. Almost half, at least half of the stuff that you talk about in chemistry is related to ions. Okay, perfect. It's an atom with some charge, right? Which just means that it has more electrons than it does protons, or more protons than it does electrons. A net charge. Exactly, Cal. Okay, so I'm going to use sodium as my example here. Sodium is often used in real life as the chemical that is used to develop this charge. Exactly, Cam. So anybody remember what the charge is on sodium? Oh, I'm going to make this smaller so it fits in here. +1. Exactly. Okay. So if this cell pumps a bunch of sodium inside of it, okay, from the blood, what you're going to get here is a net charge. Oh, this is something that you guys all know. Okay, sodium 1+. Good. If you pump a bunch of sodium, and by the way, potassium is used for this as well, which also has a 1+ charge. If you pump a bunch in here, you're going to get a net charge. Okay, which means that on the inside here, the electrical charge is going to be net positive, and on the outside, because you pumped all this sodium in, there's going to be a net negative. This is electricity. Okay, this is electricity. That's all that it is, and I think you probably have a vague recollection of that from grade 9. But an imbalance of charge is a static electricity. It's static charge, and you can absolutely detect this. There is a voltage between these two things, an electrical potential energy, because these things want to charge neutralize. So there is a pressure actually trying to force this back to neutral again. And you would have talked about this in grade 9, the idea that things want to discharge. They want to go back to 0 electrical potential energy. They want to go back to 0 volts, basically. They want to they want to become neutral. So a voltage now has been developed across this cell membrane, a voltage that you can measure with a voltmeter. Okay, this is directly connected to electricity. This is electricity. So how these cells work is that when you try and send a signal down through the cell, one part of the cell is going to release its sodium, okay, which is going to remove the polarization. We call that depolarization. We say that the cell is depolarized. Okay, because it's no longer polar. When this part of the cell depolarizes, the next adjacent part of the cell, so that would be like this area over here, the part beside it, has these pumps. These pumps right here are sensitive to changes in voltage. So when the area right next to them experiences a depolarization, a change in voltage, they also depolarize. Okay, so that means now the next pump is going to release its sodium, and so this part is going to get moved out. And on and on and on, all the way down the entire long structure of the nerve all the way down to here, and then the signal actually gets transmitted right into the body of the cell at the end of the nerve fiber. Okay, so this is sometimes called a wave of depolarization, because the wave will start at it's like a wave in a stadium. The depolarization will start over here, and then that wave of depolarization will travel all the way down the nerve fiber to the end. And that can happen extremely quickly. Over the entire body of the nerve, it could happen in 5 milliseconds, okay, which is an extremely short amount of time. And that's how we're able to basically operate like this. And if you can imagine, we're sending signals between various nerve cells in the brain. This has to happen so quickly. Imagine how quickly your brain cells have to be operating to hear my voice, see what I'm writing, process all of those things in real time, and for you to understand those things that I'm saying to you. I mean, we're talking about incredibly fast processing times. Forget about computers. This is the amount of signal that your brain is able to process simultaneously and integrate and understand is way beyond anything that we can currently do with computing. We're looking at stuff on a whole different, several orders of magnitude different than anything that we can do with computing. So I mean, these natural systems, your neural system is amazing. It's an amazing accomplishment of nature, and they're remarkably common in nature too, which is really cool. So anyway, that's the idea of nerve transduction. Okay, sending a signal across a nerve. Now, you probably are already aware that your heart is not made of nerve. You probably know that. Your heart is primarily muscle. And muscle also does this. So when you want to send a signal to the muscle, you can do this. You can have a nerve that goes to the muscle. But once it gets to the muscle, the signal has to spread out across the entire muscle to get the muscle to kind of contract together. The whole muscle, all the different cells of the muscle have to be told, okay, now we're going to contract together. So the same thing happens across the surface of the muscle, where the muscle is going to pump. So this is supposed to be a muscle cell, by the way. The muscle cell is going to pump sodium and potassium, by the way. Again, there's more than one ion involved here, but I'm simplifying it a bit. And it's going to create a polar situation here. Okay, there's going to be polarity, which just means that there's a positive and a negative end here. Okay, so the inside becomes positive, the outside becomes negative. Once that nerve signal is sent to the muscle cell right at the nerve, it's called the neuromuscular junction. There's a place where the neuron touches the muscle cell. That's going to activate the first muscle cell, and it's going to depolarize. And when it depolarizes, by the way, that actually is what activates the constriction of the muscle, that actually makes the muscle get smaller and constrict or contract. And when a muscle cell detects this depolarization in its neighbor, it will also depolarize. And then that will depolarize the neighboring muscle cell, which will depolarize the neighboring muscle cell, and it will travel in a wave across the muscle and depolarize the entire muscle. Okay, and then there's a short reset period where it's got to pump these sodium ions and potassium ions back in, and then it goes again. Then it will depolarize again and again and again. This happens many times per second, so it's a very, very rapid process. And when you do this, that you get a sustained muscle contraction. Okay, so the key here is that this is an electrical phenomenon that is happening. And when it occurs, when you actually get muscle cells depolarizing, you can actually measure this by just putting a voltmeter at the muscle. You can measure the differences in electrical potential from one side of the muscle to the other side of the muscle. You can just measure this. And this is honestly no different than the voltmeter that you used in grade 9, whatever that was. Okay? It's just a special version of that with little sticky pads that you can put on your skin. But it is exactly the same technology. There's no mystery to this. This is exactly the same thing that you've already used, but it's just a bio version of a voltmeter. Okay? So that's that is signal transduction. That is how your heart muscle is actually depolarizing. Now, there is something important to note about this, and that is that the heart muscle cannot just depolarize. It obviously has to start somewhere. Okay, that signal has to begin somewhere in the heart. And where it begins, this does go on your note, by the way. Where it begins is a spot right here in the heart. This is just, if you look on the sheet, it's just the first little heart diagram on the left. This spot right here is called the Sinoatrial Node. It's on top of the right atrium. Okay, the very top corner of the right atrium. That is where you begin a contraction of the heart muscle. So I'm going to run through the steps of this here. I'll go through this one at a time. So that little area right there on the top of the right atrium is called the Sinoatrial Node, or the SA node. Okay? We're going to talk about the AV node too. The AV node is located right here. So if we just allowed the electrical signal to pass over top of the heart in a wave, so that would be if it started up in that top corner, and then we just allowed it to go vroom over the entire heart, just travel across in a wave. Absolutely. I will absolutely post this with drawings. In fact, if you look in the completed notes folder, there's already one in there, but I can post an updated one. If you just allowed it to pass through across the surface of the heart, what would happen is you would get the right atrium contracting first, then you would get the left atrium contracting. Then you would have both of the ventricles contracting from the top of the ventricle to the bottom of the ventricle. And so the problem with this is that it would push blood in the wrong direction. If you contract the ventricles like this, what you're actually doing is pushing blood this way. And that's not where the exits are to the ventricles. So pushing blood in that direction is very counterproductive to what you want the heart to do. This is what would happen if it was just a pure wave of muscle contraction traveling over the surface of the heart. So there is obviously something else going on here. You can't do this in real life because the heart won't function. It won't pump correctly if you just essentially let the wave of contraction just pass over the heart. That is how all other muscles work. Your biceps and your triceps and whatever, they just have a wave of muscle contraction that travels over the surface of the muscle, and that's perfectly fine for skeletal muscles, but it doesn't work in the heart. So you need a special system of electrical conduction in the heart, and we're going to talk about how that system works right now, where the signal goes. Now I want to mention that this signal that starts in the sinoatrial node will periodically start all by itself. The sinoatrial node will send out approximately 70 beats per minute, no matter what. Even if it's not connected to anything, even like under all circumstances, it will just send out approximately 70 beats per minute. That's what the sinoatrial node does. So it doesn't have to be connected to your brain. It doesn't have to be connected to anything. In fact, you can take a heart. I've done this before in the biochem lab. Where you take a heart from a rat and you hook it up to some pipes. You hook up all the blood vessels to little pipes, and as long as it's getting pressure and oxygen and fuel, sugars in that case, it will just keep pumping. That heart will pump even if it's not inside of the body of an organism. It will just keep pumping. So, and by the way, we don't do that for no reason in the lab. The lab that I was working in was doing some experiments around blood pressure control medications, and they were actually testing the drugs on rats to see how the blood pressure medication affected the blood pressures that are measured inside of the heart. to see if it was safe for human use. So anyway, these were trials. These were drug trials that were being done in the lab. So it's not for funsies or anything. But anyway, that being said, the sinoatrial node is actually attached to your brain. So you have a nerve, and there's the nerve right there. I added it in, called the vagus nerve, that goes from your brain to the sinoatrial node. So there is some neural control, and that vagus nerve can be used to basically turn up the sinoatrial node, so increase your heart rate, or turn down the sinoatrial node, or decrease your heart rate. And it's really fascinating that this even works in real life. But let's say, for example, that you're walking along and you get to the bottom of some stairs. Okay, so you're just walking through the school or wherever you happen to be, and you look up the stairs. Before you even start climbing the stairs, your brain has already recognized, okay, this is on an unconscious level, that you are about to engage in additional workload. You're about to go up some stairs. You know that that's a workload. And subconsciously, your vagus nerve will actually send a signal to your heart and say, beat faster. Exercise incoming. And you can measure this. You can measure this. When people are walking around, when they get to a place where they're clearly going to exercise, and the brain registers that, your heart rate will go up before you even start exercising. You will send a signal to your sinoatrial node saying, prepare for exercise. And that's so there isn't a lag, because if you didn't basically prep your body for it, what ends up happening is you'll be 5 stairs in, going up the stairs, and there'll be a lag. You'll be behind in your use of oxygen, in your use of in your transport of oxygen. And so it just means basically that you're starting at a disadvantage when you're starting your exercise. So in order to correct for that, the brain sends a signal preemptively: be faster. We got to be prepared for exercise. So that exists. There is some neural control here. However, the neural control is not necessary. The vagus nerve can be severed fairly easily in things like a car accident. It runs this weird path underneath your collarbone. And so it has a tendency to get severed in things like car accidents. But your heart will just keep beating without the vagus nerve. You don't actually need to have your heart attached to your brain. Now, you lose some of that neural control of your heart, but it's not fatal. So your heart just keeps beating even without it. The heart's fairly important. So it's like a backup mechanism, I guess you can think of it. It doesn't need to be constantly innervated by the brain. So let me get back to what I was talking about before with where the electrical activity goes. So we start at the sinoatrial node, and what we want is to fill up the ventricles first. Okay, that's the first thing that we want. So what happens is the electrical signal is going to get sent. What color should I use for this? Maybe green. The electrical signal gets sent from the sinoatrial node down through the right atrium, and also simultaneously goes over and goes down through the left atrium. Okay, that happens simultaneously because you want to be filling both ventricles at the same time. And so both of these atria basically contract identically. They contract exactly at the same time. Then the signal travels back up to a little center spot right here. Okay, so there's a wave of muscle contraction that goes down from the top of the heart, and that forces blood down into the ventricles. So that's going to pump blood into the ventricles and fill them up. That's what the atrium are doing. And then the electrical signal is going to come to the center of the heart. Okay, that little spot in the center is called the AV node, or the Atrioventricular Node, because it is the interface between the atrium and the ventricles. So AV, Atrioventricular Node. And the reason that the AV node exists is because you need to make sure that when you send the signal to the ventricles, that you only do it once. You have to only do it once. So sometimes it'll take a little bit longer for one atrium or the other atrium to contract, and I'm talking about just by milliseconds here. And so these signals may not arrive at the AV node simultaneously. They might be off by a little bit. But the AV node puts them back together again into one signal. If you didn't do that, you would send two signals down to the ventricles, and they, instead of contracting in one smooth movement, they would twitch. And you don't want any twitching in these muscles at all. You don't want them to just be twitching. You want them to have a controlled contraction movement so that they actually push blood. If they're twitching, they're not working properly. Okay, so so the AV node basically allows the signal to come back together again, and now the signal is going to travel down the center of the heart, okay, right down to the apex. Okay, so the center of the heart, if you'll remember from our last drawing, is called the septum, right? So this electrical activity now is going through the middle part of the heart called the septum. So the electrical activity goes down through the septum. This does not cause the ventricles to contract. So they don't contract while this signal is traveling down through the septum. There's like a separate little area that this electrical signal can go down. And now what we want is for both ventricles to contract at the same time and to push blood up and out through the valves at the top of the heart. Not the AV valves, but the other valves, the valves that are going to be exiting the heart. Okay, so now this wave of muscle contraction is going to go up this way through the through the left ventricle, and this way through the right ventricle. And as it does that, this is going to be the big contraction at the bottom of the heart that pushes blood out of the heart. So that's the electrical pathway that the signal takes through the heart. And then once that's done, the heart is completely depolarized. It takes a moment to reset, and then another signal will come from the SA node and do the next beat. But it always has to travel in this very particular pattern over the surface of your heart. And if it doesn't travel in this pattern, this is where you're going to run into some medical issues, which are fairly common. But if you have an irregular heartbeat, what you really have is a problem with electrical conduction in your heart. We're going to talk about some examples of that in a moment. Where's my electrical activity? So I'm going to mention one other thing, and that is the sound of the heart. So what actually makes the sound? If you've ever put a stethoscope on somebody or yourself, we used to do this in class during this lesson, so I'm sorry to say that I don't have stethoscopes to give you at home. So you can't listen to your heartbeat, but you can look this up on YouTube if you want. But it makes a lub dub, lub dub, lub dub, lub dub, lub dub, lub dub, lub dub. And a lot of people think that the lub dub, I mean, it makes sense. There's basically two parts of the heart that are contracting, right? You've got your atrial part contracting, and then you've got your ventricle part contracting together. So it would make sense that the sound is actually just the atriums contracting, and then the ventricles contracting, and then the atriums contracting, and then the ventricles contracting. Except when the atriums contract and the ventricles contract, it actually doesn't make any sound. So that's not actually what you're hearing, although it would make sense. So what are you actually hearing then? Great question. When your atriums contract, and remember the two atriums on top of the heart contract together, okay, they're going to push blood down into the ventricle. And then there is a pause, and the blood falls back against these valves. And these valves are going to now snap shut. Okay? They actually slap closed when the blood falls back against them. That is the lub sound that you hear. So the lub is the two AV nodes slapping closed as blood falls back against them. Okay? Then the ventricles are going to contract and push blood up, okay, through these arteries. And the blood is going to fall back again against these valves right here. And these valves are going to slap shut. So that's the bicuspid and the tricuspid. When those valves slam shut, that is the dub sound that you hear. So it goes contraction, lub, contraction, dub. Contraction, lub, contraction, dub. Contraction, lub, contraction, dub. The lub and the dub are the valves slapping shut. They slap so loudly that if you put a stethoscope on your heart, you can hear the valves slapping shut. And assuming that your heart is healthy, those are the only sounds you should be able to hear going on inside of your heart. So there shouldn't be any whooshing sounds, and there should be no sounds in between the lub and the dub. Basically, everything else is silent except for the slapping of the valves. Ideally. Okay, so underneath here, I've just summarized these things. What's the lub and the dub? The lub is the AV valves slapping, and the dub is the bicuspid and tricuspid slapping closed after the ventricle contracts. I summarized some of these things that I just said below, so I'll scroll through and I'll let you write them down. And also, if you have any questions about the electrical activity or heart sounds, now would be a good time to ask them. We're going to look at an ECG now in a moment and connect them together with the electrical activity of the heart. Some prokaryote growth in there. And so just for number 4 here, I mentioned how the SA node works, which is also called the pacemaker of the heart, the natural pacemaker of the heart, because it keeps the pace of the heart kind of automatically. So those 3 numbers, 1, 2, and 3, correspond to the 1, 2, and 3 on the heart up here, just so you know. I'm just going to grab some water. I'll be right back. I'm going to let you write this. Okay, let's talk about the electrical activity of the heart. So you guys have probably seen this before, at least in basic, right? You've seen the little ECG read in the back of somebody's hospital. Maybe you've encountered this in real life, I don't know. But if you've ever been hooked up to an EKG or an ECG, and you can hear the little boop, boop in the background. These are in TV shows and stuff like that. Whenever somebody's in the hospital, they're always on an ECG, no matter what they're in the hospital for. They've stubbed their toe, they're on an ECG. It's not like that in real life, by the way. So what's actually going on in this thing, and why even hook it up to somebody? Like, what's the purpose of this? Well, if you ever look at one, and now you won't be able to unsee this in movies and TV, you'll look at it and be like, does it have all the parts that it's supposed to have? It doesn't always. There is a little boop at the beginning. Okay, this is before the big boop. Okay, a little boop like this, and notice, by the way, that this is actually measured in volts, in millivolts, but it's a measurement of the actual electrical potential difference, the voltage between the 2 probes that are put on a person. And normally there's actually more than 2 probes. You can do this with 5 probes or whatever. And it's looking at the average difference in voltage between the probes. Okay, so there are such things as 2 lead ECGs, though. There are even, in the next Apple Watch, I've read that there is going to be a 1 lead ECG, which is also possible to do, built into the watch, that can do an ECG read of your heart activity. That's really cool to me. I'm I feel like I'll get that just so I can show that to my class because I think it's cool. But anyway, it's just a measurement of voltage, okay? Electrical potential difference across the surface of the heart. So there is a little bloop at the beginning here. And what that little bloop is the atrial contraction. Okay? And so when you're looking at the ECG read, a physician looks at this and says, is this person's atria properly contracting? And there is a certain distance that this is supposed to be away from the ventricle. You can actually measure this distance, and there's a bunch of detailed analysis you can do here. But in general, is it there? Are your atrium contracting, or atria? It's possible that they're not, by the way. The atria are not absolutely necessary in order to keep you alive. They make your heart more efficient by filling your ventricles fully before they contract. But if your atria are not working particularly well, it's not necessarily fatal. Now, it can be dangerous because you can get blood clots forming in your atria and stuff like that. And so you do want them to be functioning, but they're not absolutely essential. So anyway, you check for that. Is there a little bloop before the big bloop? That's called a P wave, by the way. The letters are here. They actually have significance. So that's the P wave. Then there's what is called the QRS section, okay, or the QRS wave, or QRS complex, it's usually called. So the QRS complex, that big bloop right there, you can imagine what that is. That is the contraction or the depolarization of the big piece of muscle in your heart, which are your ventricles. And remember that your ventricles are contracting together because of that electrical system that we talked about a minute ago. So you can measure that. You can look at the depolarization of the heart, the QRS complex. Is it there? Is it strong? That is the depolarization of your ventricles. Then there's a delay. So this delay is known as the ST segment. It's right here. So it's the distance between S and T, and you can measure all of these, by the way. And there are certain numbers, lengths of time that these things are supposed to be in healthy individuals. And so you can diagnose a bunch of different heart illnesses by looking at how big these segments are. Anyway, if you're interested, I've got a whole book on it that I got from my brother. But you can learn more about this if you're interested. And then there's another little bloop at the end. Okay, so that little bloop at the end, the T wave, is the re-polarizing of the ventricles. That is, they're getting ready to go again. They're pumping the ions back across the membranes and re-polarizing, getting ready to contract again. That's called a T wave. So there's a P wave, a QRS complex, and a T wave. And assuming everything's going on in your heart that's healthy, you will have all of those, and they are roughly a certain distance apart from each other, which means that your heart muscle is conducting electrical signals correctly. Okay? So you can look at this in the hospital. When they have this, you hooked up to this in the hospital, for the most part, they're just checking to make sure that you are having a heartbeat, basically. Is your heart continuing to contract? Obviously, if it stops contracting, that's going to be very problematic for you. So that's why you're being hooked up continuously to an ECG. Now, if you're having a multiple lead ECG hooked up to you, that's when they put one on each ankle and one on your wrists, one on your chest, and then they're taking a paper lead off of it. So they're actually they're printing out a paper read of this. That paper read is being used for more detailed analysis than to just check to see if your heart is beating. At that point, they're actually measuring these segments. They're looking at the ST segment. Is this roughly the correct length? Is this roughly the correct length? You can look at these values and make sure that they are roughly within spec for a normal person. And if there's something that's way out of whack, like for example, let's say that your ST segment, that's the length of time from the end of the QRS to where your T is really long. Okay, so your T wave is actually way over here. That's called a depressed ST or a depressed T wave, when you have it shifted really, really far over to the right. And that usually means that there is there's something wrong with the resetting of your heart muscle tissue. It could be a chemical imbalance. It could be something is interfering with the repolarization of your ventricle. And obviously you get to fix that problem if maybe you have not enough calcium in your blood, or maybe you don't have enough sodium or potassium. And so then you got to do some blood chemistry analysis and figure out, is everything healthy in this person's blood? Anyway, there's a lot that goes into this. But you can use an ECG in order to diagnose a lot of these issues. So that's just the basics of ECG. Okay, you look for that next time you watch a movie or something like that. Is the ECG trace a real one that you're seeing in the background, or is it just something that was added later? A lot of people that put them in don't actually know what they're supposed to look like. So some of them are just like literally just the QRS complex and nothing else. If it doesn't have the other parts, it's not actually an ECG read. The last thing I want to talk about here is the idea of fibrillation. And you guys have heard of fibrillation before because you have heard of a defibrillator before. So what is fibrillation? Fibrillation is twitching. It's when your heart muscle tissue is not doing a coordinated contraction altogether, but instead it's just twitching. Okay? It's uncoordinated muscle contraction where you just have a bunch of random contraction happening and not a concerted pumping action. When your heart does this, it's not pumping blood. If this happens with the atria, what you end up seeing is a normal ECG read, except the atrial part, which remember is the P wave over here, ends up just being like this thing. It's just like a small twitching occurring, random contracting, instead of seeing one specific P wave. Occasional atrial fibrillation, where you have it very occasionally, is not uncommon. If you have it on a fairly regular basis, where it kind of goes in and out of atrial fibrillation, like I mentioned, it can cause blood clots and problems like that. So you probably do need to have that looked at by a physician. But every once in a while, people do get atrial flutter, basically where their atrium just go for a second. Okay, and so that does exist. If you're experiencing with any regularity whatsoever, you should talk to a physician about that. But my understanding is that if it happens every once in a while, like if you're experiencing it once a year or something like that, that's not necessarily problematic. But if you're having more consistent atrial flutter, you should definitely go see somebody about that. Anyway, it's not necessarily fatal. So you don't, obviously, we already talked about this, your atria don't necessarily have to be concertedly pumping in order for your heart to actually pump blood to your body. However, if that happens with your ventricles, remember your ventricles are actually pumping the blood out of your heart. That's the big muscular part. What you end up seeing on the ECG read is this crazy electrical activity all over the place because your ventricle is just twitching all over the place and not pumping blood. If you see this on an ECG read, it's very likely that the person is not conscious because they're not perfusing their brain with blood or oxygen. Consequently oxygen, so they're probably not conscious when this is happening. This is when you use a defibrillator. So a defibrillator is an untwitchifier. It removes the twitching, and the idea is that it depolarizes. And remember, depolarizing is when you get those ions going across the membrane of your heart muscle. It depolarizes the entire heart all at once. And so if you depolarize the entire heart muscle all at one time with a big electrical signal, you can basically, hopefully get it to reset. Then there will be a pause, and then hopefully, ideally, the SA node is going to start the process properly from the SA node and then have a coordinated contraction traveling through the heart again. That's the purpose of defibrillating somebody, shocking them, basically, to re-coordinate their heart muscle tissue. Okay? If you leave somebody in fibrillation for a really long time, eventually you're going to get this. Okay? that's when you have cessation of any sinus rhythm here, okay, at the end. In a movie, when you get this, they call, often in a movie, they call it flatlining. He's flatlining. He's flatlining. You probably heard this in a movie, I'm sure you have, or in a TV show. This is not when you use a defibrillator. So when there is no electrical activity in the heart, a defibrillator does not do anything. You can't reset a heart that has no electrical activity, because at this point, the heart is not capable of contracting anymore. It's totally depleted. It is not even twitching any longer. And so before you can defibrillate somebody at this point, you need to figure out what has caused them to go into fibrillation or flatline in the first place. What is going on chemically in the heart that it can no longer contract? Is there a problem with calcium concentration? Is there a problem with potassium concentration? Is their body temperature too high because of an extremely high fever? You got to figure out what the problem is, and you got to correct that. And then you try and get the heart to fibrillate, to twitch, and then you defibrillate the heart. But you don't use the defibrillator here at this point. And I always find this really funny, because in every TV show or movie, this is always when they get the defibrillator out. They always wait until the person has no electrical activity whatsoever in their heart, and they're like, all right, now we're going to defibrillate them at this point where it wouldn't do anything anymore. I'm sure you guys are aware that in the school we have automatic defibrillators, AED. And there are people in this school that are trained to implement them. And maybe you've taken a first aid course and you've learned how to implement an AED before. But if you put an AED on somebody and they have no electrical activity in the heart, they don't do anything. So this is not, an AED is not going to work if you put it on somebody and they have no electrical activity, because it doesn't do anything. So what it's actually detecting when it's automated, like an AED is automated, is it's looking for this. So if it detects this pattern, then it knows that it can deploy an electrical signal in an attempt to reset the heart musculature. I just thought that's really interesting to me, is that it seems like Hollywood, well, certainly has no idea how a defibrillator works, but they never deploy it at the correct time. Watch for this in TV shows and movies from now on, and it will be so annoying to you, because they will always deploy the defibrillator when it doesn't do anything anymore, almost every single time. I just threw this in because I had an ECG done not too long ago, and this is my ECG tracing. So I just wanted to show you what it looks like in real life, and you can see my P wave and my QRS complex, and then there's the Re-Polarization at the end. I thought, I better take a picture of this. So that and by the way, that's a healthy ECG. I have no problems with the electrical activity in my heart. So that's just a normal ECG tracing. Okay, how am I doing for time here? Oh boy, I think I talked a little bit too long there. What I want to talk about really briefly now. Oh, I just did some drawings underneath here too of what atrial fibrillation looks like. It's when you got a bunch of extra stuff. You can still see the QRS complex really clearly, but then the other components in between are kind of obscured by the twitching of your atria. So I want to talk really briefly about other mechanisms that your body uses to increase or decrease your heart rate. So you may have one time been on a roller coaster, or is it possible for you to go skydiving? I don't know if you're old enough. Maybe not. You've done something exciting, something that has made you go, or maybe you watched a horror movie and it really scared you. Okay? Any of those things could potentially activate what's called your fight or flight response. Your fight or flight response is a neurological response in your body, a physiological response that is activated by danger. And when you are in danger, and you don't necessarily have to be in actual danger, you just have to be in perceived danger, like when you watch a horror movie and you're scared, that's perceived danger. The stress response that your body has in response to that is that you produce adrenaline. So you have adrenal glands, they're sitting on top of your kidneys. and they produce a chemical called norepinephrine. Norepinephrine is also known as adrenaline. I should mention that norepinephrine is also called noradrenaline. So there's 3 words there: noradrenaline, norepinephrine, and adrenaline. Those words all mean the exact same thing. Why are there 3 words for the exact same thing? I have no idea. But they do all mean the same thing, and they are used interchangeably with each other. So norepinephrine, adrenaline, and noradrenaline are all the same chemical. This is the exact same chemical that is found in an EpiPen. If you have an EpiPen, if you've got severe allergies, what you're trying to do with your EpiPen is activate your fight or flight response, which has this really cool blood vessel dilatory effect, constriction effect, the exact opposite, in that it will prevent blood flow into the periphery. And so if you're having extreme swelling due to blood vessel dilation, let's say in your neck and you can't breathe, if you take some norepinephrine or some epi, it will cause constriction in those blood vessels and redirect blood flow away from the swollen area. And it's almost instant. It's so fast, and it causes a huge reduction in swelling in those areas pretty much instantaneously. And it allows you to breathe again if it's causing obstruction of your airway, which is kind of cool. So that is an adrenaline response. As part of that fight or flight response, that adrenaline also can bind to your SA node. So remember, the SA node is an area of your heart that controls how fast your heart is beating. And basically, when epi binds to your SA node, it tells it, turn this puppy up. We're in danger. You need to increase your heart rate right now. And so it does. So your heart rate will increase. It could increase quite a bit, actually, because of interactions with epinephrine. So that system of preparing for danger is called your sympathetic nervous system. Okay? So the sympathetic nervous system is basically used for dealing with stress. I'm talking about extreme activation of your sympathetic nervous system. That would be when you feel like you're in physical danger, when you are afraid, when you are feeling the thrill of being on a roller coaster. That's actually activation of your fear mechanism, basically, your sympathetic nervous system. That's a strong activation. You can also get a weak activation of your sympathetic nervous system when you just experience everyday stress. Are you worried about getting that assignment in on time? Are you worried about passing that course? Are you worried about your driver's test? Are you worried about somebody in your family for whatever personal reason? Worry and stressing about things also has an effect of activating your sympathetic nervous system. And so there are consequences to activating it long term. If you're always stressed, if you're always worried, you can get a buildup of hormones called cortisol. Cortisol is what your body produces to deal with long-term activation of your sympathetic nervous system. And there are a bunch of side effects of having high cortisol in the blood. Over the short term, it's basically helping your body deal with stress. But over the long term, if you have a constant high cortisol level, it can cause all kinds of problems with metabolism. And I'm not going to get into it. It's not directly related to this course, but you should look it up. Having elevated cortisol for an extended time period is problematic, which is one of the reasons why it's so important to have proper stress. What's the word I'm looking for here? Having a good not a routine, but a method, having a method in your life for dealing with life stress. Are you taking time out of your day to meditate? Are you doing something to help manage your stress in your life? Exercising, having a good diet, all those things help deal with stress. Because you do want to inhibit your activation of your sympathetic nervous system so that you're not always on stressing about things. Because in the long term, that cortisol pathway is going to cause problems for you, damage to your circulatory system, etc. So anyway, something to mention here. But anyway, in the short term, sympathetic nervous activation increases your heart rate. Okay. You also have the opposite system of this. Okay. When the stressful situation passes, and hopefully it does, you activate what's called your parasympathetic nervous system. Okay. And then that releases a chemical called acetylcholine. So the acetylcholine is actually released in the junction between the nerve and the muscle. So it would be where the vagus nerve meets the SA node. So we talked about the vagus nerve coming from your brain to the SA node. Where those two things interact with each other, that's called the neuromuscular junction, the junction between the neuron and the muscle. And that the neuron actually releases a neurotransmitter. That's a chemical for sending signals between nerves called acetylcholine. And acetylcholine has the exact opposite effect of adrenaline, which is that it causes your heart rate to decrease. It's basically turning off your fight or flight response. And this the parasympathetic nervous system has more of a gradual effect, as opposed to the sympathetic nervous system, which has a very rapid onset effect. Escaping from the lion is an emergency. Relaxing after you've escaped from the lion is not an emergency. So it's not quite as a rapid deployment there of the parasympathetic system, but it basically does the opposite. So that is another mechanism that can be used to control your heart rate. I got two more things I'm going to talk about here, and then we're going to talk about some illnesses. Cardiac output. Cardiac output is a measurement of how many times your heart pumps per minute. Not how many times, sorry, how much volume your heart pumps per minute. So the volume that your heart pumps is different at rest than it is at exercise. So one way that you can increase your output is by increasing your heart rate, right? You can just pump more times. The other way that you can increase your cardiac output is by increasing how hard your heart pumps. So your heart actually pumps harder when you're exercising. It's pushing harder on the blood. It's not just pumping faster, but it's also pumping harder. So you can turn that up to some extent. That increases what's called your ejection fraction, which is the amount of blood that is actually pushed out of your ventricles. So when your ventricles pump, they don't always pump 100% of the blood out of them. And the percentage that they actually pump out of them can go up when you're exercising, the ejection fraction. So you turn that up when you're exercising. The last way that you can improve your cardiac output is by actually strengthening your heart muscle. So you can increase your ejection fraction by making your heart stronger. And the way that you make it stronger is by exercising. So the more cardiovascular exercise you do, which is like sustained endurance exercise, the stronger your heart muscle gets. It gets bigger and it gets stronger. And so you're able to pump more blood per minute. We talked about that previously. It also means that your resting heart rate goes down because you don't need to have as many beats in order to pump the same amount of blood. So that's how it relates to fitness. Basically, you improve your cardiac output the more fit you are. That's not the only way to increase oxygen transport in your circulatory system, but it is one of the ways. You also increase your amount of red blood cells. You increase how much your blood vessels are integrated into your muscle. And there's a bunch of different methods, which we're not going to talk about in this course, but increasing your cardiac output is part of that mechanism. And the last thing that I want to mention is the idea of blood pressure. So whenever you have a fluid going through a system of tubes, you're going to have pressure. Pressure is basically how hard the tubes are pushing against the flow. Okay? And there's going to be some pushback, obviously. When you turn on your water in your hose in your backyard, the water pressure from your house is going to push water into the hose. And the more the hose pushes back, the higher the pressure is going to go. If you've ever covered up the tip of your hose, you may have noticed that the flow decreases, so less water will come out of the hose, but the pressure is higher because you've restricted the flow. Okay. If you're interested in fluid dynamics, I've got some great engineering videos that you can watch. But anyway, that's the basics of it. So you can measure how hard your circulatory system is pushing back against the pressure from your heart. Okay, that's your blood pressure. It's how much pressure there is in your arteries. Okay, and what you're trying to approximate here is the actual pressure in the left ventricle. That's the part that's pushing into your blood, but you can't measure at the ventricle. It's too difficult to measure at the ventricle. So where you typically measure is the closest artery that's near the surface to the ventricle. Okay, and actually technically that would be your carotid artery on your left-hand side. However, you're not going to put a blood pressure cuff around your neck and cut off the blood supply to your head. That would make you go unconscious. So that's not a great place to take blood pressure measurements. So as an alternative, we can very briefly cut off the blood supply to your arm, and we can use that to make blood pressure measurements, which is exactly what we do. So normally in the course at this time, I would have you measure your blood pressure. We have some cuffs. We can't do that, so I'm just going to talk about the process. So you take a cuff and you put it around your upper arm, and you're going to inflate that cuff until the pressure in the cuff, which you can measure, is going to completely cut off the blood supply to your arm. And that's why it's a little bit painful when you get your blood pressure measured, because you do have to inflate the cuff enough that there's no flow through your brachial artery, through the artery that's going to go into your arm. Okay? And then you're going to slowly release the pressure from the cuff until blood starts to flow. Okay? That point at which blood is going to start to flow means that the pressure in the artery is the same pressure as the pressure in the cuff. Once those two pressures are equal, that's going to be your maximum amount of pressure that you can make in your artery. Okay? And so you want to know that maximum pressure. That's the pressure when your ventricle is contracting and pushing hard on the blood from your heart. So that pressure while your ventricle is pushing is called the systolic pressure. And whenever you write a blood pressure measurement, the systolic pressure is the number on the top. Okay, it's usually written as one number over another number. And it's measured in millimeters of mercury. I mentioned that that is a typical unit of measurement for pressure when we're talking about living systems. I don't know why. It's a leftover from early biology, European biology, but we use it now. It's ingrained. So millimeters of mercury, I could measure that in kilopascals too for you. I could do the conversion, but 120 millimeters of mercury is how you're always going to see it written, which is known as the average or healthy systolic blood pressure. And there's obviously a range here. So that pressure that you see during the contraction of the ventricles could be a little bit lower, it could be a little bit higher. Maybe it's 100/80 for you. Maybe it's 125/80. Those are all normal measurements. So if you've ever had your blood pressure taken, chances are for somebody at your age, you probably have a normal systolic blood pressure. Okay? Now, the pressure never goes down to zero in your arteries. Even when the ventricle is not contracting, there is still a leftover pressure from the last push, basically. So the lowest pressure that you achieve in your arteries is called the diastolic pressure. This is basically the pressure while your ventricles are resting. Okay? If the pressure went down to zero, you'd end up getting a halt of blood movement in your arteries, and you may even get backflow of blood in your arteries, which you don't want. And so the pressure never actually goes down to zero, but it goes lower when the ventricles are relaxing. So while they're relaxing, that you measure the diastolic pressure. The diastolic pressure is what is written on the bottom of a blood pressure measurement. And a typical diastolic pressure is 80 millimeters of mercury. That's typical. Could be a little bit lower. If you're an elite athlete, it may be lower. It doesn't need to be very high in order for during relaxation, in order for you to pump all the blood again, because your heart muscle is more fit. It's able to pump more blood. You don't necessarily need a very high diastolic pressure. So this number could be a little higher, a little lower, but it's usually somewhere around 80. The last time I measured my blood pressure, it was 122/83. So again, I have a very typical blood pressure. You probably do as well. If your blood pressure measurement is way out of those numbers, that would be an indication that you may want to chat with a medical professional. Maybe there's nothing to be concerned about, but certainly if it's way outside of that number, you may want to investigate that. Now, there's one caveat here, and that is the typical blood pressure at rest, when you are relaxed, okay? When you're exercising or stressed, that number can be considerably higher. And so you have to be very careful, even as a physician, when you're measuring blood pressure. You have to remember that people are often nervous when they're going to the doctor, or at least some people are. And so sometimes you get a little bit of bias there. People are nervous, their blood pressure goes up because they're nervous. And so you're actually measuring like an anomalous blood pressure reading of going to the doctor blood pressure and not blood pressure at rest. So often if a doctor is seeing high blood pressure measurements, but they suspect that it may be because you're nervous in the doctor's office, they may give you a blood pressure monitor that you wear like 24 hours. You take it home. And then they can get a better idea of where your resting blood pressure is at. So it's not always a great indicator to take it at the doctor's office because some people are stressed when they go there. So something to consider. But you don't actually have to have that high of a blood pressure in order to be considered having high blood pressure or hypertension. Hypertension, by the way, is the word for high blood pressure, hypertension. The problem with having too much pressure in the vessel, which is any systolic pressure over 144 or any diastolic pressure over 94 at rest, is that over time, those pressures actually damage the blood vessels. It's fine for them to be higher during exercise. In fact, it's healthy for your blood pressure to go up considerably during exercise. It just isn't healthy for you to live like that at rest. So anyway, it does cause problems damaging the blood vessels. We're going to talk about some of the consequences of damaging your blood vessels in a minute. But anything over 144 over 94 is problematic. And it's if either of the values are above that. So if your, like I said, if your systolic is above 144 or your diastolic is above 94, that's generally considered to be chronic hypertension. And so it may actually require medication for you to regulate your blood pressure at that point. So just something to mention. And that brings us sort of to the end here of our discussion of the physiology of the respiratory and circulatory system. If you're like me and you find this stuff to be absolutely fascinating, when I did this in high school, this was I found this absolutely fascinating. This was probably one of the most interesting things I did in high school was learning about the physiology of body systems and things like that, which is why I chose the degree that I did, which was kinesiology in my undergrad. There is so much more to be learned about this. If you're interested in athletic performance, if you're interested in biochemistry, there's so much deeper that you can go. And so I first of all, take grade 12 biology, especially if you like biochemistry. But if you have any interest in biology at all at the academic level, you should definitely take grade 12 biology. And you should consider learning more about this at an institution of higher learning, a university somewhere or college. I started on track for medicine at the beginning of university. My brother did as well. My brother is a physician. And then towards the end of university, I was not a huge fan of the lifestyle that physicians typically live, which is extremely high stress. And it does not really leave a whole lot of family time. I suppose it depends on the style of physician that you are, but certainly my brother was under a lot of stress. So I switched out of the pre-med track. I was in the pre-med track. And I decided to go, well, first of all, I worked at a biochemistry lab for a couple of years, but I moved out of the out of the medicine track, and I decided to go into teaching later. Part partly because when I was in grad school, the teaching part was the best part for me. I loved teaching classes in grad school. And you get an opportunity to do that as well. You'll get to probably teach some courses if you are taking a master's. Oftentimes as a TA, you get to teach, which is fun. So anyway, that was the best part of me when I was doing my master's program. And so that was what led me into teaching. Actually, that's when I switched over to teachers college. But my brother decided to stay on the medicine track. But anyway, this stuff is the most interesting stuff to me. And there's so much more to be learned. The reason I'm mentioning this is you're just skimming the surface of this stuff, and there's just so much more. So if you're interested, it's out there. Let's talk about some illnesses of the circulatory system. How am I doing for time? I got 10 minutes. Okay. I don't want to cut into the time after the break too much, so I'm going to I'll be relatively brief with these, because I do want to give you time to work on your project. But keep in mind, if you see anything you're really interested in here, you could make one of these illnesses potentially your focus for your project, because you're going to pick a technology, and you're also going to discuss at least one disease in detail. And this could be one of these. So a disease that interacts with your technology. All right, so we talked about all this stuff. The first illness I want to mention, and remember that on the sheet what you're looking for here in general is causes, symptoms, and treatments, but it's more of a fill in the blank here. I tried to simplify this sheet a little bit, so it should follow the presentation here fairly closely. So what's a heart murmur? You've probably heard of this before. Well, it's just a sound. We mentioned that when the heart is pumping normally, the only sounds that you should ever hear are the lub and the dub. Lub dub, lub dub, lub dub, lub dub. But if you hear something else, I'm just gonna grab my wife's stethoscope here. Family full of physicians. Let's see if I can hear it. My wife works from the basement too. This is a nice one. Yeah, that sounds good. Just a lub and a dub. If you hear anything extra in there, that's generally a cause for concern. So it does matter how much extra you hear. So you can grade the audibility of the extra sounds. Like for example, if you hear a lub, sh, dub, or lub, dub, sh, lub, dub, sh. Some extra stuff. What you're hearing is turbulent flow. That's flow of blood that is kind of whooshing around inside a ventricle or an atrium. And what normally makes turbulent flow, there's two main things. One, you can either get backflow through one of your valves, so your valves are not watertight, basically. Or you could have a hole, and this is actually shown on the bottom here. You could have a hole in between the left ventricle and the right ventricle through the septum. Either of those is possible. When you're born, you actually everybody has a hole between the left and the right ventricle. But the moment you start breathing, that hole begins to seal. So the reason why the hole is there is because before you start breathing, you don't actually need to be pumping blood to your lungs, because your lungs aren't doing anything when you're in the womb. So you're basically bypassing your lungs by going through the hole in the center of your ventricles, which is totally fine. But once you are using your lungs, then you don't want any blood to go in between the two, because otherwise you're allowing it to avoid going through your lungs, which you need to oxygenate your blood. So you need to seal the hole up. If it's a teeny tiny hole, and there are very small murmurs, grade 1 murmurs, little tiny ones that aren't super problematic. I mean, obviously a physician is going to make that determination, but it may require no treatment. But if you have a larger hole, you're going to end up probably needing that to be surgically repaired to make your heart efficient at pumping blood. And the same thing goes for the valves. If there's leaks in the valves, they're very small. Sometimes it's not a big problem. If they're moderate or fairly large, you may require a valve replacement. So there's various ways to do that. You can get artificial valves. You can get a pig implanted valve that's from a pig. They have very similar cardioanatomy. That's an excellent question. Let me ask my wife. I didn't actually know that, but I want to know the answer to that too. I bet my wife knows. One sec. Hey, love. That's handy having a physician around. My wife mentioned that's probably a better question for my brother, who's an anesthesiologist, but the basic answer to that question is when you put somebody under anesthesia, it's a stress on their heart. So it's not that significant of a stress. However, if you have a heart murmur and the cause is not necessarily known, you only want to put people under full anesthesia if you're very confident that their heart is functioning properly, unless you absolutely have to, like you're doing a procedure that will absolutely require full anesthesia, for example, a heart transplant or something like that, in which case there's no way around it. But if you can avoid it, you don't want to stress the heart too much, because there can be unpredictable consequences of that from a general anesthesia. For example, if you're depressing heart function a little bit with general anesthesia, you may cause an unusual sinus rhythm, electrical activity in the heart or something like that, if you don't know with confidence that their heart is functioning properly. That's the basic answer. And for a more detailed answer, I'll have to ask my brother. He could probably give you the textbook answer. But by the way, as an aside, would anybody be interested in a short presentation from a physician? I can't guarantee which one, but either my brother or my wife, talking about how to get into medical school, the sort of the path into medicine and what it's like to be a physician. Some people asked in previous years about this, and they've done presentations before. Both of them have, my brother and my wife, because students have a general interest in this, or at least sometimes they do. If people would be interested, let me know. Okay, cool. Even if it's just a few students that are interested, we can definitely do that. I'll have to ask them if we can arrange that. So I think I missed a question here. Would you be able to feel it? Do you mean feel it in yourself, Lillian? My understanding, I mean, if it's ventricular fibrillation, the person is going to be unconscious because they don't have coordinated beating of their heart. And when you feel their pulse will be very erratic. So you can feel it through their pulse. They won't have a regular lub-dub pulsing that you feel in their pulse. It'll just be basically like or it'll be no pulse. In which case, you're going to initiate, as a person who's providing first aid, you're going to initiate this, which is called, oh Lord, CPR. Sorry, I don't know why I couldn't think of that right away. Cardiopulmonary resuscitation, CPR. so you're not really concerning yourself that much with whether or not you're detecting fibrillation. That's somebody else is going to have to make that determination with an ECG. But the person themselves, if you're experiencing atrial fibrillation, just atrial, you can perceive that as a flutter in your heart. Like you can potentially feel it, but not always. Atrial fibrillation is often goes undiagnosed and people don't notice it, so it's not always obvious. And oftentimes you have to give people a long-term ECG that they would wear around to monitor their sinus rhythm throughout the day to try and detect things like atrial fibrillation. Is that right? Yeah. It feels irregular when you feel your pulse? When you have AFib? Okay, there you go. You want to come over and talk about the heart rhythm?
I was just gonna say with anesthesia, you're like artificially inflating the lungs, right? So your heart has to work harder to overcome that pressure a little bit.
Oh, that's interesting. A little bit.
So that contributes to why.
I got to let these guys go for lunch right now, but cool. I'll mention that. I'll mention that when we come back. Anyway, I'm gonna let you guys grab something to eat. There's not much left here, so this will just take only like 15 minutes when we come back, and then I'm gonna give you the rest of your time to work on your projects. Okay. Anyway, enjoy your lunch. Welcome back. Please let me know in the chat if you are here for the second half, if you haven't already done so. Let's pick up where we left off. So we had discussed heart murmurs, this idea of extra sounds. Any extra sounds are technically bad, although how loud they are is important. If you have a grade 6 murmur, I wanted to mention this, I just didn't get to this before we finish. It's actually loud enough that you can probably just hear it, like in the room. So that would be a very severe murmur. So it is possible for it to be like pretty significant. But again, obviously treatment depends a lot on severity. My wife wanted to point out again with regards to the question of the anesthesia, the idea that when you're under anesthesia, your lungs are being artificially respirated for you, right? Ventilated externally. And having that positive pressure on your lungs actually creates an added stress on your heart. Again, it's not that significant, but because of that additional pressure that's pushing against your heart, again, you avoid doing it if you're not confident in the heart's ability to handle stress, unless you absolutely have to, in the case of something like a heart transplant. But anyway, MI. So this is the one that a lot of people, I think, are aware of. People have heard of a heart attack before. Up until maybe the last 5 years, this was the most common cause of death. Heart attack and stroke were the most common cause of death. Cancer has slightly overtaken heart attack and stroke as the most common cause of death now in the last 5 years. That shouldn't worry you. It's not that cancer has become more common. It's actually because we're getting better at treating MIs and strokes. So if you're better at treating them, they become less fatal. And so something's going to kill people. You have to die from something eventually. And so then the next most likely thing is cancer after an MI or stroke. So it's not necessarily an increase in the prevalence of cancer, but a decrease in the prevalence of dying from heart attack and stroke because we're a little bit better at treating them. So that's over the last 5 years. So but it used to be the most common way to die. That's an uplifting thing to talk about. But anyway, something's going to kill you at the end. So basically, it's where you interrupt the blood supply to your heart. So I don't mean the blood supply going through the superior and the inferior vena cava and into your right atrium, or the blood returning from the lungs going into the left atrium. When I mean the blood supply to the heart, I mean the blood supply that is actually traveling through the arteries of the heart. So those are these arteries right here. So those are called the coronary arteries. This was mentioned in the video. Those coronary arteries run backwards from the aorta. So you can see that aorta is coming out of the heart right here, and there's a little bit of a back flow. So every time the ventricles rest, you actually get backflow a little bit into these blood vessels that are called the coronary arteries, and they feed the heart muscle tissue. And as you can imagine, as a muscle that is contracting all the time, there is a fairly large oxygen demand, a blood demand from the heart muscle tissue. And if you block that blood flow, and we're going to talk about some mechanisms for blocking later, but essentially a plug falls into one of those passageways, and they're already narrowed, usually due to, again, stuff we're going to talk about in a second. You cut off blood supply to an area of the heart, the muscle tissue is going to start to die, and not instantly. And the longer the blood is obstructed, the more tissue dies. So generally, what's going to happen is you're going to get a blockage, and you're going to start to experience pain, usually experienced on the left-hand side of the body more than on the right-hand side. You get arm pain and chest pain. Also you can get it in the neck on the left side. Shortness of breath, nausea, vomiting, sweating. People get anxiety or feelings of anxiousness that are associated with it as well. It's very uncommon in young people, by the way. You probably don't need to worry about having a heart attack, but after the age of 50, it becomes significantly more common in the population. And as I mentioned, it's caused by a blockage in your coronary artery. If you get immediate medical treatment, oftentimes you can be given drugs, and the main treatment here is to get clot-busting drugs, which are chemicals that break clots apart and hopefully will break up whatever clotted matter has fallen in and blocked your coronary artery. If you do that fairly quickly after the time of obstruction, usually you can recover most of the function from the muscle that's been damaged. But obviously, the longer that your blood is occluded or restricted from flowing to a particular area, the more damage this is going to occur. So, and obviously, if too much damage occurs to your heart muscle tissue, then you die. So you can die from a heart attack. Certainly, people do die from them if they're severe. And how much blockage is also important there. If you have a fairly significant blockage, and by the way, this is not done while you're having a heart attack. We're not talking about removing the clot that is blocking the passage completely. That is usually done through drug therapy, through a clot-busting drug. However, the reason that clot was even able to plug up your coronary arteries is because you have plaque that has started to line the inside of your coronary arteries. This stuff happens over time due to poor diet and lack of exercise. We're going to talk more about that in a bit. But the vessels are essentially ripe for plugging because they're already significantly narrowed due to this cholesterol plaque buildup. So what you can do is you can run an extra pathway, an extra vessel around the blockage. So that's a strategy. The blood vessels are usually taken from the leg. You usually take veins from the leg and use them to build pathways around the blockages. So that is called a bypass, and you can have a double bypass or a triple bypass. It all depends on how many bridges they're building around the blockages. And it's fairly effective. So this is a fairly common procedure. Obviously, you have some disease mechanisms in place already that have led to your current blood vessels being occluded. And so if you continue in the same lifestyles that have led to that occlusion occurring in the first place, the bypasses don't necessarily last that long. But this certainly relieves some of the immediate problem. Now, there are other ways to fix the occlusions. You can use lasers to blast away that plaque. You can use essentially tubes that you put in and expand inside of the blockage to kind of push the artery a little bit wider to allow blood flow. That's called a stent. And there's all kinds of stuff that they do with stenting, like they're slightly radioactive, which prevents new clots from forming on top of them and stuff like that. However, those are fairly temporary. And also, if you blast away a lot of the blockage or drill away the blockage, keep in mind that that blockage usually goes somewhere. And one of the possibilities is that it could cause a blockage somewhere else, like in your brain, and cause a stroke, which could kill you. And so those types of surgeries are not as common as they used to be. They are still done, and it all depends on which modality of surgery is going to provide you with the best outcome. So if it's not going to be a good outcome potentially through a bypass, then they may try some other technique. Here's a picture of somebody who's had some bypasses done, and you can actually see there is some extra vessels there on the heart that don't belong. Those are bypass vessels. When I did a human dissection as part of my first year human anatomy course in university, they do that at Waterloo if you're taking kin. Not every university has a human anatomy lab, but the person that we dissected had several bypasses, which are visible on the heart, which is kind of cool. And they also had an artificial pacemaker, which was kind of cool. So we actually saw when we dissected the heart and stuff, there was a pacemaker there, which is fascinating. Some of you I know are doing the pacemaker as your treatment technology for the project, which is kind of cool. But anyway, that's what it looks like. I also just want to mention, sort of as an aside here, that if you are removing the heart, and this is an open heart surgery right here, where you would do a bypass, or do a bypass, right? Exactly. Or potentially you could do maybe this is more involved cardiac surgery, like a heart transplant or something like that. You can see that the steel retractor right here is actually holding the ribs apart. That's this device right here. You got to break the ribs because they are covering up your heart, and then retract them using this device, push them apart, and give you a nice little work area to work around the heart. And obviously, this is a last resort. This type of surgery has a fairly long recovery time. You're busting somebody's chest open. There's a significant possibility of infection and stuff like this. Post-op, you lose a fair amount of blood. So the risks are moderate. We tend to favor surgeries that are laparoscopic, so they're done with instruments that are inserted through small holes that have tiny cameras attached. We tend to favor those whenever possible because they have a faster healing time. There's less opportunity for infection. There's lots of benefits to laparoscopy. But some things you just simply got to crack open a chest for. So this would be an example of this surgery right here. On the side, this is a heart transplant operation that's happening. On the right-hand side, that big old machine is a heart-lung bypass machine. And so while your heart is removed from your body, as happens in a heart transplant, and while you're working on the heart, you can't really work on it while it's under high pressure. If you've ever, and this may not be something that's ever happened to you, but if you've ever tried to replace the nozzle at the end of a hose while the hose is turned on, you may have found that it's quite difficult to put a new nozzle on the end of a hose while there's pressure in the hose. And that's because obviously the pressure is resisting your attempts to close things and the same thing is happening inside the body when the heart is pumping. So if you're going to be working on a pressure vessel in the heart, you have to turn the heart off. And you can't just leave the heart off, or obviously the person will die. So you need a machine that is going to do all of the work of the heart outside of the person. And that's what that heart-lung bypass machine is. It's the machine that's over here, and there's a person operating it. This guy right here is the perfusionist. And you can do that full-time, especially if you work in a larger hospital. I don't think we have full-time perfusionists in KW. I think you need to be at a larger hospital, but there would be someone that does that type of job here. Certainly in Hamilton or in Toronto, there'd be several. Anyway, a heart-lung bypass machine has to do the work of both, right? Because remember that your heart is not only pumping blood to your body, but it's also pumping it to the lungs, and those are separate circuits. So the heart-lung bypass machine is doing exactly that. You can't leave somebody on a heart-lung bypass machine for a really long time. You might think, well, why don't you just leave a person on that until you can get a transplant, for example? So just have this operating in the background. Well, first of all, when you run blood cells, living cells, through artificial tubes, they do get damaged a little bit. Red blood cells don't like to be pumped through plastic tubes. It stresses them. And so over time, you're going to start getting damage to your blood cells that are being pumped through this machine. And the pumps obviously cause a little bit of damage as well. I mean, they're specifically designed to minimize that, but you still get a little bit. And also, your blood vessels require that pulsing motion that comes from the heart. They are specifically adapted to receive that pulsing pressure from the heart. And it's very, very difficult to artificially replicate that in a way that responds to all of your body's needs. Remember that the heart can change its rate of pumping, and it's all based on the hormone signaling and signaling that is coming from the rest of your body. And so it's working in tandem with your blood vessels to provide adequate blood for various tasks. And so if you don't maintain that close communication between those two things, over time you're going to start getting damage accumulating, which is exactly what happens when you're on an artificial heart or on a heart-lung machine for a very long period of time. So it's not a long-term solution, but it certainly works during something like a transplant surgery, which is kind of cool. Stroke. A stroke is basically the same thing as an MI, a myocardial infarction or a heart attack, except it's just happening in your brain. So there are two types of stroke. There is one where it's just like an MI, where there's a blockage in one of the blood vessels in your brain. That's called a ischemic stroke. Ischemia is the name when there's lack of blood flow to an area. So an ischemic stroke is a stroke that is happening that is causing lack of blood flow due to a blockage. So that would be when you have a clot or a piece of material that's in your blood that has caused a blockage to a part of your brain. There's also the other kind of stroke, which is where you get a breakage in a blood vessel in your brain. That's called a hemorrhagic stroke, and you get bleeding that's happening in your brain, which also prevents proper blood flow and perfusion of blood into an area of the brain. So both of those cause lack of blood flow to an area of the brain, but they're basically the opposite of each other. So one is a bleed in your brain, and then the other one is a clot in your brain. So because there are these two types of strokes and the symptoms that you get from the two are basically identical, it's very important to figure out what is actually causing your stroke before you start treating it. Because if you have a blockage and you treat it by, imagine this. The treatment for a blockage is you got to break the blockage, right? You got to put a drug in that prevents clotting and breaks clots up. So if you gave that drug to somebody that had a rupture that already has bleeding in the brain, it's going to make the bleeding much, much worse and kill them. Okay, so that's obviously problematic. And if the opposite happens, if somebody has a rupture in their brain, what you want to do is you want to give them drugs to increase clotting. You want drugs that are going to help repair the damage in the brain. And if you give those to somebody who has a blockage or who has a big clot, it's going to make the clot much worse, which will kill them. So essentially, if you treat the wrong type of stroke, you're going to kill your patient. And so it's very important that you figure out what kind of stroke is going to happen before you attempt a treatment. So that's normally why you get a really fast. If you're coming in with stroke symptoms, they're going to give you a CT scan, which is basically a three-dimensional X-ray of the brain to figure out if they can find any clots in your brain. So if anybody's doing CT, stroke is a cool one that you could potentially do as your illness to talk about. So what happens when you don't perfuse your brain cells with oxygen? We already talked about this when we talked about the respiratory system. They die very, very quickly. So brain cells are extremely sensitive to lack of oxygen. And anything that your brain does, which is a lot of different things, can When it doesn't do those things, that could potentially be a symptom of a stroke. So for example, your brain is responsible for voluntary muscle movement, for moving your muscles around. Well, if you experience paralysis, then that's that part of your brain not functioning. In other words, there's a lack of blood flow to that area of your brain. That is a symptom potentially of a stroke. Did you suddenly lose your ability to speak? Well, your ability to speak comes from your brain. So it's potentially a symptom of a stroke. Now, death obviously would be the most severe version of that. If you block off too much of your neural activity, it's going to cause brain death. But that would be in the most severe circumstances. There are lots of other potential symptoms that you can find. And just think of everything that your brain does. It allows you to, see colors and identify objects and have memories and talk and be able to understand things that are said to you. And so the symptoms of stroke can be all kinds of weird stuff. Did you suddenly lose your ability to understand what numbers are or something like that, which happens, by the way? That's potentially the symptoms of a stroke. So you have to think about all the different things that the brain could potentially do. There are common lists of symptoms that are associated with certain regions in the brain not receiving enough oxygen, but you can get all kinds of weird ones depending if you have small areas that have blood occlusion. Would blood thinners be something that would decrease clots? Yes, that's the purpose of taking them for the most part. So people that have are at high risk for heart attack or stroke, they're generally on blood thinners as a precautionary measure to prevent the formation of clots that could potentially cause a stroke, or a heart attack. So yes, that's a treatment for that. A blood thinner is essentially preventing clotting, is what it's doing. For the project, do we focus on one illness that our technology diagnosis treats? You can talk about multiple, but you only need to talk about one in detail, Jaleel. And talking about one in detail is perfectly acceptable. That's all you need. Some people like to talk about a couple if there's a couple closely related illnesses that the machine is specifically designed to treat. That's fine if you want to talk about a few, but you only need to talk about one. Anyway, I mentioned this, that obviously they can range in severity. You can have a tiny little occlusion to a small part of your brain. Those are called microischemic events or microstrokes. That exists. Sometimes they're transitory, which means that you just momentarily have an obstruction and then it goes away. Those actually might be very common. We don't have a lot of good data about that because people often don't get diagnosed with that. But transitory ischemic events, which are like short-term blockages that resolve very quickly, may be actually very common. There's some evidence to suggest that they're probably a lot more common than we know. But anyway, we only really care more about the events that are prolonged, that are causing damage to the brain. So it's the second most common cause of death after cancer. So cancer is the most common, stroke is the second most common, and heart attack is currently the third most common. And it is a very common cause of disability. So when you see people that have, especially older people, that have a lack of mobility, paralysis on one side of their body, stuff like that, it's often caused by stroke. The burnt toast myth. So the burnt toast thing is associated with a heritage history minute that was aired widely in the late 90s about, I believe the doctor's name is Walter Penfield, who was one of the pioneers in neurosurgery. And the burnt toast thing has nothing to do with strokes whatsoever. And the history minute is all about actually the treatment of epilepsy through neurosurgery, not strokes. But I will say that smelling and perceiving smells is one of the functions of the brain. So having an unusual smell perception or losing your smell perception can potentially be a symptom of a stroke. Now, losing your perception of smell can be associated with a whole bunch of different things. So it's not a clear indicator of stroke, but anything that the brain does could potentially be a stroke indicator if it stops functioning. Now, the idea of having a specific smell with a stroke, I've never heard of that. I think that's a complete invention. But the burnt toast thing specifically is down to that Heritage History Minute, which I should post for you so that you can watch it. But that was actually associated with a person having an epileptic seizure or having unusual brain electrical activity, which was resolved through neurosurgery. Has nothing to do with the stroke whatsoever. So its short answer to your question is that is a myth. Yes, that has nothing to do with stroke. So what are the things that potentially cause stroke? It's the same things that could potentially cause a heart attack. Hypertension, which we just talked about, high blood pressure. Smoking, which damages the lining of your arteries. Diabetes, which also causes damage to the lining of your arteries. And high cholesterol, which enhances the damage that is caused to the inside of your arteries. And we're going to talk about that damage in a moment, but those things are all associated with higher risk of stroke. So just really quickly here, an ischemic stroke is, again, a blockage. And when you look at a functional MRI, which is what's on the left right there, that's an MRI image that takes into account the actual use of the tissue, which is really cool that you can do that. There is an area of the brain that is experiencing no use. It's not functioning in the MRI. It shows up as a dead zone in the MRI, which is it's dead because it's not receiving any oxygen. So you can detect that with a functional MRI. MRIs are time-consuming, and so they're not usually used in an emergency situation unless you cannot diagnose it with a CT scan, which is where you're going to start. But they can be used for emergency medicine as well. So that's ischemic. Hemorrhagic is the opposite, and this is easily detectable on a CT scan because you can see areas of the brain where there's blood where there shouldn't be blood. So there's basically a pool of blood somewhere in the brain where there's not supposed to be blood there. And so you can see that on a CT, and you have to, usually they do an injection of dye into the blood that shows up on the CT, and then you can see with the dye where the blood is bleeding in. It allows the blood to show up on the CT scan. And normally blood is not particularly obvious on a CT scan, but it's a radioisotope, I believe, that they give you in your blood, and then it makes the blood really visible. Will the diseases be a major part of the quiz? No, it's not a major part. There are questions about the illnesses, but they're fairly it's a fairly minor part. It's more about structure and function. The quizzes are more about structure and function. I'm just saying there's no questions. There are questions on illnesses, but it's mostly a focus on structure and function of the three systems. The different sounds. That's a great question, Michaela. It's a complex device. It's rotating. There are a bunch of servo motors that are rotating a giant magnet around your head or whatever body part is being scanned with the MRI. And most of what you're hearing is like the rapid activation and deactivation of the servos that are moving the magnets around, or there are large solenoids. Solenoid is basically a electronic switch that is turning on and off the magnet. It's sending a very strong magnetic pulse. For anybody that's doing MRI as their technology, it's a very strong magnetic field that is being suddenly turned on and suddenly turned off. And just in very brief here, it causes all of the atoms in a particular sample to line up with each other with the magnetic field that's being placed across it. It's mostly hydrogen atoms. And then the atoms, when you remove the magnetic field, they fall back to their original position. And when they do that, they give off an electrical signal. An electromagnetic signal, which you can detect. So you can use a detector to see the signal that basically comes off of the material after it falls back to its initial state. And so you have to turn that magnet. It's an extremely powerful magnet, very high voltage. You're turning it on and off, on and off, on and off. And so the solenoids that switch it on and off, they make a sound when you turn them on and off. And that's the sound, most of the sound that you're hearing inside the MRI. So you got some solenoids, and you got some servo motors that are rotating stuff around, and that's the majority of it. It's quite loud. I had one fairly recently, and they give you earplugs. It's pretty loud. Yeah, so and again, there's a whole bunch of different rotating magnet arrays inside, and they all have their own system of servo motors, and it's a real complex thing. So, my understanding is that it's mostly servo motors and solenoids that you're hearing. But it's a very complex device. There's obviously other components in there as well. Just most of the electronic stuff actually is silent. So I'm fairly confident that that's making up the majority of the noise. But there's probably other aspects to it as well. That's interesting. Yeah, I had one recently too. It was very loud. So, okay. These two, often confused, because if you look at their names, look at that. Really similar, right? Arteriosclerosis and atherosclerosis. So one of these is the hardening of the artery. That's the arteriosclerosis. So the hardening of the artery, but by the way, these things are closely associated with each other, and so sometimes these terms are actually used interchangeably, but they refer to a slightly different process that is occurring. Okay, so the arteriosclerosis is a calcium buildup on the inside of the artery. It basically makes the inside of the artery crispy. Okay, and so when I did my human dissection, and we like held the aorta and stuff like that in our hands with gloves, you can actually feel, especially in the elderly, especially if they're not very healthy, that they are crispy. The arteries are actually hard. And over time, that calcium has been built up on the inside as a response to damage that has occurred to the surface of the artery. And this is associated with all kinds of things. I mentioned that high blood pressure causes the damage to the inside of your artery. Smoking causes damage to the inside of your artery. Diabetes causes damage to the inside of the artery. So all of these mechanisms, lack of exercise over time will cause damage. Having an extremely high cholesterol, having high cholesterol blood, which may or may not be associated with diet, depending on your genetics. But having very high blood cholesterol over time causes damage. So anyway, all of these mechanisms are causing a buildup of calcium, and underneath the calcium, a fatty deposit is occurring. So the fatty deposit itself is the atherosclerosis, but the calcium on top is arteriosclerosis. And together, they are referred to as sclerotic plaques or plaque in your arteries. So both of these things kind of put together is referred to as plaque. And sometimes you'll just hear people say, well, this person has a lot of plaque in their arteries. It's actually a combination of arterial and atherosclerosis. There are slightly different mechanisms at play for both of them, but they are similar mechanisms and they both narrow your arteries. So the calcium buildup on the inside is arteriosclerosis, and the fats that build up underneath are atherosclerosis. So basically cholesterol, LDL cholesterol specifically, which is low density lipoprotein cholesterol, which is a protein that is used to transport fat in your blood. It's one of the proteins that's used to transport fat in the blood. Ends up depositing some of that fat underneath the surface of your artery. Okay. So it forms a fatty area underneath the surface. And again, the fat is getting deposited because of a rupture in the artery that is caused by, and again, all of those things that I mentioned: lack of exercise, poor diet, smoking, diabetes. Those are probably the main causes, are causing damage, which is allowing this fatty deposit to be built up. And then it forms a fibrous cap, and the fibrous cap is partially responsible for the calcium buildup that's happening over top. But all of these things together are causing your arteries to become narrowed. Okay, they're basically building plaque on the inside of the artery and causing a narrowing of the artery. So what happens when you narrow your arteries like that? Well, first of all, you can't vasodilate and vasoconstrict very easily. And it's very important to be able to do that because that's how your body regulates where it's directing blood in your body. For your organs and stuff that are built off of extremely large arteries, like your internal organs, it doesn't affect them as much, although it is really hazardous for the kidneys specifically. Your coronary arteries, obviously if you get these plaque buildups in your coronary arteries or in your brain, it's going to make them very susceptible to a clot forming and potentially blocking the artery. So that's going to make you very susceptible to a heart attack or a stroke. If you have these narrowings of the arteries occurring in your arms and legs, essentially you cannot pump enough blood to your arms and legs in order to functionally use your muscles and to keep your skin alive. So if you've really narrowed the arteries that are going to your arms and legs, eventually you're going to start getting necrosis that's happening in your arms and legs. It's very painful. It's like a burning sensation or pain. And then that's quickly followed by a warning, gross slide coming up ahead. Actual necrotic damage happening to your peripheral tissue. So it could be happening in your toes or your hands. And again, this is a result of damaging your arteries and getting arterial and atherosclerosis, which prevents proper blood flow to your extremities. The number one cause of this is uncontrolled diabetes. So in uncontrolled diabetes, especially in the peripheral arteries, causes a lot of damage and restricted blood flow to your extremities. I'm sure you guys have heard of amputation before. Both of these people are going to have an amputation, almost certainly, because they have far, far too much necrotic damage, and it's going to require amputation. Most amputations in North America are a result of diabetes. I don't want to say all. Obviously, people have accidents. You get run over by a train or something like that. Or maybe you have a cancer or something that requires a amputation. That does happen. But by far, the most common cause of amputations is actually from diabetes. So both of these are diabetic, by the way, in origin, which is unfortunate. When I say diabetes, I mean uncontrolled diabetes. So in other words, you have type 1 diabetes and you're not properly regulating your blood sugar over a long time and taking insulin when you need to be taking insulin. Eventually that's going to cause severe damage to your arteries. Or you have type 2 diabetes, and over time you're not taking measures to control your type 2 diabetes. Type 2 diabetes is usually lifestyle related, although not always. But it's related to body mass and things like that. In both cases, you do need to make some effort in order to control your blood sugar. Otherwise, having chronically high blood sugar is going to lead to arterial damage. Hypertension, we already mentioned this. This is the idea that you just have too much back pressure pushing back against your heart beating for a long period of time. It's going to cause damage to your arteries, and it's also eventually going to end up in heart failure. If you have really high blood pressure for an extended period, it starts to damage your heart's tissue. Essentially, there's just too much pushing back against your heart pushing out. You get some weird thickening in your ventricles, and this is not the hypertrophy that you get from exercise. It's a different type of hypertrophy. Hypertrophy just means increase in size in the muscle. And over time, you're going to start damaging your lungs because you're pumping with too much pressure into your lungs, which damages the thin barrier between the capillaries in the lungs and the alveoli. So you may get fluid retention happening in your lungs, and eventually it just leads to heart failure, which is essentially not being able to pump enough blood to your body any longer, which can lead to death. So in the very long term, hypertension can be fatal because it is eventually going to cause some types of cardiomyopathy, some disease mechanisms that are going to happen in the heart. But long before that, you're going to be experiencing problems with your arteries and arterial and atherosclerosis. We mentioned this idea of measuring blood pressure just very quickly here because we already talked about this. There are two parts to the blood pressure measurement: systolic, which is when the heart's contracting, and diastolic, when it's relaxing. And a normal pressure is 120/80, and that's in millimeters of mercury. Next time you're in the drugstore, I don't even know are we allowed to go to the drugstore? I never leave the house anymore, so I have no idea what people are allowed to do. But if you go to the drugstore, a lot of drugstores have a blood pressure monitor that it's just, you can just put your arm in it. It's just like for people to test out. I don't know if this is like I don't know if you're supposed to do that with COVID. If you're supposed to use a blood pressure machine. I don't know. I'm not sure actually. Maybe check with the pharmacist before you jump on that thing. But anyway, you can just check out your blood pressure, see what it's at. This last little bit here on timekeeping we already mentioned, so I'm gonna rifle through this real fast. I want to give you time to work on your project. So SA node, we already mentioned, is the pacemaker of the heart, and it works together with the AV node to keep everything in electrical timing. An artificial pacemaker is when your SA node does not do a great job of that. So for whatever reason, you're getting arterial fluttering, as I mentioned, atrial fluttering, atrial fibrillation, or maybe occasional ventricular fibrillation, which would be very serious. And every time that you get that unusual electrical activity in your heart, the pacemaker is basically like a defibrillator. It's going to give you a small electrical signal to the heart, which will reset the beat of the heart. These things used to be gigantic when they were first invented, and they worked on lead-acid batteries, which are gigantic and run out of power fairly quickly. They're not rechargeable, etc. You used to wear this outside of the skin, and then there would be like a little interface on your skin where this would go underneath. Now they're fully under the skin. They're quite tiny because you can make a really powerful, long-lasting rechargeable battery that actually sits underneath the skin. And you just have to put like a recharger that sticks magnetically to it on the outside for a little bit of time. I don't know if you have to do it daily. Probably not. It's probably weekly or monthly. And you have to recharge it every once in a while to make sure it's operating correctly. You can even send Bluetooth hardware updates to it or software updates to it and stuff like that. There's all kinds of new technology for these now. But anyway, they're used to treat arrhythmias. So if you know that you have atrial fibrillation and it's significant, it's happening fairly often, or maybe your heart is beating too slow sometimes, or maybe it's beating far too fast sometimes, or irregularly, often those problems can be managed with an artificial pacemaker. So it's cool that we have that technology. There's an example of one right there. That's a fairly old model. It's pretty big. They're quite a bit smaller than that now, which is kind of cool. And they run the leads up through a large vein. That's a brachial vein, I believe, up there, and then wraps around and goes through to the inside of the atria or ventricle, depending on the type of pacemaker you have. So the leads can go to different places depending on the type of pacemaker that you have inserted. Okay, I believe this is the last thing. Oh no, varicose veins is last. I better hurry up here. I want to give you time. Carbon monoxide poisoning. CO, carbon monoxide, not carbon dioxide, carbon monoxide, is very, very sticky to hemoglobin. It sticks to hemoglobin much, much better than oxygen sticks to hemoglobin. So if you bind your hemoglobin to carbon monoxide, every time that your red blood cell makes a circuit, instead of picking up new oxygen and dropping off CO2, it basically is just transporting carbon monoxide in a circle. And depending on how many red blood cells you have stuck to carbon monoxide, there are various degrees of seriousness here to this carbon monoxide poisoning. It takes a long time for carbon monoxide to unstick to a hemoglobin. So if you have, for example, 25% of your hemoglobin bound to carbon monoxide, that's essentially removing 25% of your oxygen transportation capacity from your body. And it doesn't matter if you remove yourself from the source of the carbon monoxide, because it stays stuck to your hemoglobin for a very long time. So obviously, if you bind enough hemoglobin to carbon monoxide, it's going to be fatal, because you're going to suffocate. Even though you may no longer be exposed to carbon monoxide, there's no way to remove it from your hemoglobin, and so you could potentially suffocate. That's why some people die from carbon monoxide poisoning after the fact. So they've been exposed, now they've been removed to fresh air, but you can still potentially suffocate. So it's the scary thing about it is that it's produced in pretty much every combustion reaction. There's always some being produced. It's colorless, odorless, tasteless. It's not an irritant. And so you will not know that this is in your home unless you have a carbon monoxide detector, which are required by law now in your house. So you probably have one. Certainly every building code requires them now, at least in North America. So you probably have carbon monoxide detectors already. But So the main issue here is that it can be a byproduct of combustion from a furnace or a fireplace or whatever. And so if you don't have a detector, if this happens to seep into your home, which is physically possible, you won't know until it's probably too late. So that's why the detectors are so important. Obviously, if you're not transporting enough oxygen in your body, you're going to get some symptoms from that: drowsiness, confusion, dizziness, headache. But those are not necessarily alarm bells for people. If you're feeling tired or maybe a little bit confused, drowsy, you have a headache, some people just assume that that's like a cold or something like that. So it's not that obvious that it's being caused by carbon monoxide, which is one of the reasons why it's so scary. So how do you treat it then? What do you do? Well, you can give people 100% oxygen. And then if you do that, it's more likely that the carbon monoxide is going to detach from the hemoglobin and be removed from your blood. It's not perfect, so it doesn't instantly cause all of the carbon monoxide to be removed, but it certainly increases the rate that the carbon monoxide is going to be transported back out of your circulatory system. So you can start with that. You can also do a really cool thing, which is you can put somebody in a hyperbaric chamber, which is basically oxygen under pressure. So now you're increasing the amount of oxygen, but you're also increasing the partial pressure of oxygen, right? Because you're increasing the total pressure that the person is being exposed to. Going back to the partial pressure thing that we talked about in the respiratory system. So the total pressure of oxygen is going to go way up if you're in a hyperbaric chamber, which is going to force more oxygen into your circulatory system. And it's also basically gives a greater pressure for CO to diffuse out of your circulatory system. So hyperbaric treatment is probably the gold standard for carbon monoxide poisoning, but we don't have a lot of hyperbaric chambers around. They're pretty much used for this exclusively and for treating diving injuries. So they're not super common. There is some hyperbaric therapy in Toronto, but I don't know of a local hyperbaric treatment facility. It's also used for treating burns. Can help for burn treatment. I don't think we have any in KW. Hamilton Hospital has a hyperbaric chamber, I believe, and there are several hospitals in Toronto that do, but I don't know if we do here. Anyway, this is just more here about carboxyhemoglobin. This idea that CO is going to stick to the hemoglobin and prevent binding with oxygen. Okay, last thing I'm going to mention is this idea of varicose veins. We mentioned that veins have valves to prevent backflow. And this is just a normal vein here with a valve that's preventing blood from flowing backwards. If you have blood that is, for a very long period of time, placing great pressure on the valve, in other words, I've never heard of that, Ryan. That's possible. I've heard of oxygen therapy being used for lots of things. Now, I don't know about the efficacy of that. It may have no efficacy. In other words, it doesn't actually do anything. But certainly, it's possible that people are attempting that. Hyperbaric therapy of oxygen, like actually putting somebody in a pressurized chamber of oxygen, is extremely expensive and difficult to do. If you're using anywhere near the types of pressures that would be used for therapeutic use, that chamber is very expensive. So I would guess that people are not using that to treat anxiety, but maybe using 100% oxygen therapy for something like that is possible. I don't know about its efficacy. I've never read anything about that, so I won't speak to its efficacy. But it's certainly possible that it's being used for some type of psychopharmacological therapy. I'm very skeptical. I'm highly skeptical. I'd like to see a paper on the efficacy of that. But anyway, it's possible. If you are standing for long periods of time and you have lots of back pressure, especially in the legs, on the veins. Interesting. Yeah, celebrities do a lot of stuff, and I don't think that they investigate the efficacy of a lot of the things that they're doing. They're spending millions of dollars on, so they might not even care. But I highly dubious. I'd like to see that study that they've done on anxiety and hyperbaric therapy. I'd love to see it. I suppose it's possible. certainly the cost benefit analysis on that is going to be very dubious. But anyway, okay, you got lots of back pressure here. Let's say you're standing for long periods of time every day. What tends to happen is that you get sort of this bulging of the venous material around the valve, and eventually you can actually start getting the body building offshoot veins here in an attempt to find a pathway for this blood to be returned to normal venous flow. And this is more common in any condition that's going to cause your connective tissue. Remember that the outside of these veins are made of connective tissue. If you have any situation where your connective tissue is being made more flexible, and a great example of that would be pregnant women. So when you're pregnant, as a result of the change in hormones in your body, you are intentionally making your connective tissue more flexible. And the idea being, obviously, you need to push a baby out of you. And so as part of that, as part of trying to push a baby out of you, you do need to make your connective tissue more flexible to allow that to occur without significant damage. And so that does happen. Your connective tissue actually does get softer. And it can allow you to develop varicose veins. It also happens more commonly to women in menopause. And again, that's a hormonal thing that's involved there. However, this can happen to anybody. Men get it too, but it's much, much more common in women, especially during pregnancy and post-menopause. It also tends to happen in people that are abdominally straining a lot. So if you don't get a lot of fiber in your diet and you're doing a lot of pushing, you may end up also with developing something like varicose veins. So this is what it looks like. Again, you get these bulges and offshoots that are developing in veins. For most people, this is not painful and requires no therapy. People don't like it because it doesn't look great, or at least subjectively it doesn't look great. Maybe some people don't care, or I mean, certainly if you're older, I don't think it's that big of a deal. But again, people have different standards for that. You have to make that determination yourself. So people often get therapy cosmetically to remove them because they don't think that they look good. But if you're not experiencing pain, they don't usually require any therapy. Now, some people do get pain from them. They can be painful, in which case therapy makes a lot more sense. But there are obviously potentially side effects from it. What the therapy usually is they do an injection into the varicose vein, the bulge, the offshoot of the vein, basically, and that causes that part of the vein to die. It gets blocked off, it dies, and it gets reabsorbed. I'm not sure exactly what chemical they use for it. It'd be interesting to find that out. But anyway, you can also have them surgically removed, so you can have them tied off and snipped out. Obviously, there's complications associated with that as well, so there's no perfect treatment for this. But most cases of varicose veins don't actually need any treatment because they're not painful, and so it'd be more of a cosmetic treatment. Okay, there's my little blurb on illnesses of the circulatory system. Any questions about those before we move on? That's the last thing I'm going to say, by the way. We're done. We're done with the unit. It's the last thing I'm going to say about systems. Ryan, let me know if you find anything about hyperbaric oxygen therapy, especially for anxiety. I'm very curious. Especially academic sources. So what are you working on now? Well, let's pull this up so you can see it. If you'd like, you can watch the fetal pig dissection as part of a review before you write your quiz. Totally up to you. I leave it there for you as an option. It's recorded from last quad. Other than that, there are no questions to go along with this. We kind of just did the questions together. I'm trying to save you time. You basically have the rest of this time to work on your medical technology presentation. Remember that everything is coming due tomorrow at midnight. If you'd like a little bit of extra time to work on stuff, please just write me and let me know what your alternate deadline is for yourself. And I'm happy to approve those as long as the deadline is reasonable. Okay? But please let me know if you need more time. Try not to bump stuff too much. because you're going to start running into deadlines for the next unit. The next unit is also very busy. It's not like we hit a lull where you'll suddenly have time to finish all this stuff. So just be aware of that. So shoot for tomorrow at midnight for all this stuff. There's always a quiz, medical technology assignment for this unit, but there's always an assignment and the vocabulary list. So it always comes due midnight of the day after the end of the unit, which would be tomorrow in this case. Okay, again, if you need more time, give yourself a reasonable alternate deadline, email me and ask about it, and we'll discuss that deadline together. Will you have time to work on the project tomorrow? I don't think so, but let me check. Kind of, sort of. So actually, whoa, it's a super light day tomorrow. So there's a single lesson. It's a little bit long. It's 44 minutes long. But there's a single lesson. I've pre-recorded it to help keep things on track. And then there are 4 questions that go along with that lesson and an exit quiz. And that is everything for tomorrow for both blocks. So actually, yes. The short answer to your question is yes, there is time to work on this tomorrow, because you will, that will only take you maybe an hour and 15 minutes, hour and 20 minutes max. And that's the entire day tomorrow. So, so yes, you will have time to work on this tomorrow. I'm sure the last time running through the course, I had more on this day. And then I think this is probably the spot where people start to feel backed up. Project wise, and so then I must have cleared a bunch of stuff off this day, which makes perfect sense. Okay, I'm gonna stop talking. I'm gonna let you guys work. If you need anything, looking for sources for your project, have questions about the project, anything at all, please let me know. I'm gonna be working on getting your midterm reports together, and they will go out at some point today. I don't know when, but whenever they're done, they will get pushed out to you. There'll be an email to you and to your parents. I'll see you tomorrow.